Women in Data Science Worldwide has been around since 2015. We started pretty small, as just one conference on Stanford campus, and our aim then was to simply feature outstanding women doing outstanding work in data science. And then we grew. We grew into a global movement, connecting women and also men around the world.
WIDS has grown organically, responding to what the needs of the community are, what women data scientists need, and exploring new ways to interact. Stanford University is proud to have been the birthplace of WIDS, and a decade of growth. With our unique strength in data science, computation, and AI, Stanford was the ideal incubator to support this force for advancing diversity in data science. WIDS has had a lasting impact on data science at Stanford, with a co-founder of WIDS as a founding member of Stanford Data Science. Including multiple perspectives is baked into the core identity of our programs.
It's critical, and we need women in these roles. Every individual brings their own perspective, and so we need to make sure the entire workforce is represented.
Because it really fosters that sort of community building you want when it comes to not just data science, but really any, I think, efforts to try and engage more girls and women.
So the fairness mission that we have is really try to enable that two people with equal talent should have an equal shot at opportunities.
I also just encourage people not to give up and keep arguing for what you believe in and what's important to you and what you think will make a difference.
WIDS is about the sense of urgency we should all feel to ensure that women earn that seat at the table based on their skills and talent across the world.
WIDS has inspired me to believe that more is always possible.
WIDS has inspired me to think big. WIDS has inspired me to learn more.
It brings together some of the leading scientists in the world.
The journey is just beginning. At WIDS, we're not just envisioning a more inclusive future in data science and AI, we're making it happen. From equipping students with critical skills to advancing career development, we're expanding access to education, mentorship, and empowering support for tangible impact. The next decade starts now, with action, with us. Join us in shaping the future. Volunteer, mentor, or donate today.
Now that we are all excited, I would like to welcome you and thank you for joining us today for our 7th annual Women in Data Science at Lane Medical Library conference. My name is Boglarka Huddleston, and I am the manager of research and instruction here at Lane Medical Library. I would like to start with a few housekeeping items. We do have a Q&A box at the bottom of your screen, so you can ask questions throughout the conference. If you see a question in the Q&A box that you like, please push the like button, and then it will move up. It will basically upvote it, and we'll make sure that we'll get to those questions. Live captioning is available for the duration of the conference. Just click the CC icon at the bottom of your screen to view the captions. I would like to very quickly cover the agenda for today. We will begin with opening remarks from Colleen Cuddy, who is the Director of Research and Academic Collaboration here at Lane. Then Dr. Tina Hernandez-Boussard will deliver the keynote, the opening keynote. After her talk, we will transition to our panel discussion and meet our 3 panelists. I will moderate the discussions. We do have some ready-to-go questions, but we'll also take questions from the audience. Then we'll take a 5-minute break and wrap up the conference with Dr. Cecilia Lee's closing keynote address. The conference is being recorded, so if you need to step away, you will receive the recording via email once the event concludes. As a gentle reminder, we do have a code of conduct, and thankfully I never had to enforce it or refer people to it, so I'm hoping to continue with that tradition. But you can see the code of conduct on your screen, and if you have any questions or issues that I need to address, please contact me after the conference. I would like to thank the planning committee for their dedication and hard work in the last 6 months to ensure that this event is a success and that we can continue into the future. Colleen, Amanda, Connie, and Danielle, you are appreciated more than you know. I would also like to now share a short video this is a welcome video from our Director of Research and Academic Collaboration. She cannot be here today, but I have a short video from her. So let me start that.
Hello, and a very warm welcome to Lane Medical Library's 7th Annual Women in Data Science Regional Conference, WIDS at Lane. I'm Colleen Cuddy, Director of Research and Academic Collaboration at Lane Medical Library, and I'm delighted you have chosen to spend part of your day with us today. I apologize for not being able to be with you in person, but I have a family obligation that prevents me from being here, and I look forward to catching up with the recording later this evening. Over the next 3.5 hours, we'll explore our 2025 theme, Shaping Data-Driven Medicine, Women in Health Informatics. Together, we'll examine how women are harnessing data to fuel clinical innovation, improve patient outcomes, and reimagine what equitable, evidence-based health care can look like. Ooh, that was a tongue twister. Each spring, this conference has marked the evolving frontier of data science and medicine from a woman's perspective. Last year, we focused on artificial intelligence and health equity, spotlighting the responsibility we share in building ethical, inclusive data sets and technologies. That conversation set the stage for today's deeper dive into health informatics, moving from why equity matters to how data-driven practices transforms care at the bedside, in the clinic, and across populations. We're going to kick off today with an opening keynote from Dr. Tina Hernandez-Boussard, whose work on the intersection of biomedical informatics and health services research shows data's power to improve quality and safety. After a dynamic panel moderated by our colleague Boglarka Huddleston, we'll then close with Dr. Cecilia Lee, whose groundbreaking ophthalmology and AI research reveals how the eye can serve as a window into systematic health. I know that we have many returning attendees to this event, and I'd like to remind everyone that WIDS is more than just a single event. It's a growing global network that amplifies women's voices, sparks mentorship, and invites all of us to keep learning. So today I encourage you to connect in the chat and on social media using hashtag WIDSLane2025. Throughout the year, please explore WIDS worldwide programs, such as datathons, podcasts, and upskilling workshops. And finally, as you leave, bring today's insights back to your team so that data-driven medicine benefits every patient everywhere. I'd also like to take a moment now, before I leave you, to thank our speakers, panelists, and organizers for making this event possible, and then to each of you for bringing your own interest, attention, and expertise to this very important topic. I invite you to engage with our speakers, ask bold questions, and leave inspired to push the boundaries of what's possible in data and health informatics. So with that, a warm welcome to WIDS, and I will turn it over to Bogie. Thank you.
And acknowledgement on the screen. And with that, I would like to it is my honor to introduce our opening keynote speaker, Dr. Tina Hernandez-Boussard. Dr. Hernandez-Boussard is an Associate Dean of Research and Professor of Medicine, Biomedical Data Sciences, Surgery, and Epidemiology and Population Health at Stanford University. With a rich background and vast knowledge and expertise in biomedical informatics, health services research, and epidemiology, she is at the forefront of advancing healthcare through the development, evaluation, and application of innovative methods. Through her research, she aims to effectively monitor, measure, and predict equitable healthcare outcomes. By leveraging real-world data, her team works diligently to construct a solid body of evidence that can significantly enhance patient outcomes, streamline healthcare delivery, and provide valuable guidance for health policy decisions. In addition, Dr. Hernandez-Boussard focuses intensively on mitigating bias and enhancing equity within artificial intelligence applications and healthcare settings. Through her research and evaluation of AI technology, she seeks to advance healthcare practices while ensuring that diverse populations receive equitable resources, care, and outcomes. She's here today to give us the keynote, the opening keynote. Tina, welcome, and thank you for joining us. And I will stop sharing. So please take it away.
Okay. Can you see my screen?
Yes.
Wonderful. Well, thank you so much for this opportunity. Thank you to Women in Data Science for having this meeting. It's a great pleasure to be here today and kick off this meeting. I want to talk to you today about how we really think about building responsible AI for inclusive health solutions. And let me just tell you where I'm coming from, right? So just a little bit about me. I am an Associate Dean of Research for the School of Medicine. And under that role, I'm the director for our Health Informatics core for the Clinical Translational Science Award at Stanford. I also lead the informatics and data resources for the School of Medicine and advise on health AI. In addition, I'm a professor of medicine where I have a vibrant team of diverse workers where we really think about how do we optimize real world data? How do we develop and evaluate tools that are responsible and inclusive across the entire healthcare ecosystem? And in addition, I have a third role. I'm an advisor for the National Institutes of Health for the director for artificial intelligence in health and informatics needs. So I'm coming to you wearing these different hats, and you'll see that throughout my talk this morning. I try and really pull in these three different roles, which really drives my focus, my research, and what I'm going to present today. So let's get started. Let's get started. There we go. So we know there's a growing role of AI in health and healthcare, right? AI is transforming healthcare at an unprecedented pace. From predictive analytics to diagnostic tools, AI is now deeply embedded across the continuum of care, right? Its applications span clinical decision support, healthcare operation efficiencies, and clinical trial design and operations, right? This landscape is underpinned by advancements in machine learning, natural language processing, large language models, and it's trying to make our healthcare system smarter, faster, and more precise. However, there are concerns about the advances in AI and the guardrails we have in place for ensuring that what we deploy in health informatics is safe and equitable. So my team's been taking a holistic view of the AI lifecycle, right? It's important to understand the tools we develop, how equity and biases can enter across the AI lifecycle, and how do we ensure the responsible use of AI, right? Bias is introduced at any stage of the AI lifecycle, and it propagates, it amplifies downstream. That includes data creation and data acquisition. So where are we getting the data that we use to train our models? Who is included in those data? And importantly, who's not included? It's about the choices we make in model development. Are we using a random forest, an XGBoost? What types of algorithms are we using, and why are we deciding on one algorithm over another? Model evaluation is a huge piece of the AI lifecycle. Oftentimes, we see teams focus on model performance without thinking about how we think about the fairness of the models, how we think about these models might be applicable to different populations. And then we also often have less attention to model deployment, and I'll talk about that this morning in my talk. Real world implementation and the use of the AI model at point of care, with the clinician, with the patient, and bias can enter at this life cycle at any point in time. So it's really important we think about that and think about how we develop responsible AI across the healthcare system. And one of the key points, I think, is AI has really been developed in the past for the convenient population. What does that mean? Well, data diversity is not systematically evaluated. Model deployment is understudied. And the policies and standards and guidelines for actually using AI in healthcare are limited. And we've been thinking about this and writing about this for several years now. Because it's important when we train our models on the majority population, there's other populations where they can incur harm. And when AI fails for marginalized groups, we don't just get inaccurate predictions, we actually amplify real-world harm. And true leadership means recognizing that equitable AI isn't just a bonus feature. It isn't just a checkbox. It's the minimal viable product for any healthcare application. And true diversity means centering our applications across all the populations for which they will be applied, right? And so that brings us to our first critical juncture, the data itself. And I want to talk about three aspects of responsible AI today. I want to talk about data and data representation. I want to talk about AI deployment and clinical utility. And then I want to talk about the policies and standards that are in place to actually move forward with AI in healthcare. And this, of course, is all based on this AI lifecycle that I just spoke about. So let's start with data. It's important to understand that data is never neutral, right? Data is a social artifact. Healthcare data sets are a reflection of historical priorities, historical means. So when we use these data to develop tools, we're not just analyzing data, we're inheriting social practices. And many of these past practices might not be acceptable today. So it's important to understand that data is not objective, right? Every data set mirrors who society values. Here's an example of a census data from 1980, where you can really see the way they're recording race really highlights this hierarchy of what was acceptable in that time. And so your training data is really like a political document, right? What we're capturing, who matters in that data set. And so we've recently been doing some work in the genetic space, and we've been thinking about data representativeness. So, and as I mentioned, healthcare data often lack diversity, right? And this convenient sample that I mentioned, it's mainly male, urban, insured patients. So when we use that data to develop our AI models, we're learning the patterns from these urban insured patients. And patterns of healthcare access, patterns of treatment are very different in this population compared to other populations, rural communities, underserved communities. And so by thinking about data representativeness, we need to think about when we're using data to train our model, who's included and who's not included. And so we have several ways of thinking about this. And in genomics, this has been really amplified in how we've lacked diversity in collecting genomic data. And so we need to ask ourselves, what types of diversity matter? In order to address this, we need to think about what are the goals of the questions we're asking? What's the tool I'm trying to develop? And how do we understand the data needed to ensure that the tool aligns with ethical norms? What types of data are missing from my convenient sample? And then we need to think about how do we plan to achieve types of diversity in data? It's very difficult to capture data. We know that. That's why we go to this convenient sample. So how do we think about the implications of the choices made? I always say that Stanford University, we use a lot of our Stanford clinical data, and it's not very diverse. That doesn't mean I'm not going to use Stanford data. Of course, I can learn so much from using Stanford data, but I have to be responsible in the way I use it. I have to be transparent about who's included in that sample, and more importantly, who's not included. How can I learn from that data and apply those learnings to other communities? And as I mentioned, we've been thinking about this more and more in the genomic space and design and discovery, right? Because we know there are gaps in genomic data representation that are becoming increasingly critical in AI-driven drug discovery. So this is some work we published late last year in Science. We have some another follow-up study coming out, hopefully this summer. And here we developed a novel large language model for genetic code. Our approach involved two distinct training phases, first on eukaryotes, then on prokaryotes. And this dual model architecture enables us to accelerate three key applications: drug discovery, target gene modifications, and synthetic genetic code generation. But crucial to this design decision and this work was about the human reference genome. So the human reference genome is very biased. I believe it contains over 80% of European ancestry, and it's missing over 20% of African ancestry. So what does that mean? That means when we use the human reference genome for drug discovery, for biomarker discovery, we're really doing this for a specific population, a population of European ancestry. So to ensure global applicability in these tools that we've been developing, we deliberately excluded the human reference genome from training. And instead, we looked for evolutionary distant species. So this population-free approach, which we validated through rigorous testing, shows comparable performance across all human ancestries. So what's key here, this was only made possible through our collaborative framework by really thinking about who's included in our population and who might benefit from this work. We brought together a team of diverse perspectives and scientists from the start, and we've created tools that work equitably across populations, which is really necessary. Because when these populations are missing from our training data, when we build tools that are not equitable across populations, not only do we see a hit in performance across populations, but we also end up over-predicting or under-predicting an outcome for these populations. Here's some work we did looking at adverse outcomes at the start of chemotherapy among cancer patients. And what we found was that our tool worked fairly well across racial and ethnic minorities, but we really failed for Medicaid patients. And so we really had to think hard, what's different about Medicaid patients compared to private insured patients or Medicare patients that's making our model not work accurately for these for this population? But we really had to think about it. We had to study it, and it takes a lot of extra time. And so this work, in addition to our EVO2 work, really exposes the fundamental tension, right? We're really pushing innovation, but we need to balance that with equity and generalizability across populations. And this brings us to this topic about data proxies that we often use in clinical informatics, right? And so data proxies is when we don't collect a variable of interest that we need, right? And so instead we use a proxy variable of interest. So for example, we might want to model care need, but we don't really collect that variable. There's not a variable that really captures who needs care and who doesn't need care. So oftentimes we use cost of care as a proxy. We've seen this, there's a lot of work been looking at when we want to capture criminal behavior, and instead we capture arrests as a proxy. Well, we know that arrests in populations are associated with more patrolling, more policing, not necessarily a direct correlation with criminal behavior. So let me take this a little further in some work we've been doing, looking at Black mothers' delivery and outcomes in the US, right? So we know that in the US, Black mothers have significantly higher rates of adverse birth outcomes compared to white mothers. This includes maternal mortality, preterm birth, low birth weight, and infant mortality. And this also, we also see that Black women have higher rates of cesarean deliveries compared to white women. This is a graph here from the March of Dimes from 2005 that really shows these rates, the average rates in the US from 2020 to 2022. And we see this bar right here, that middle bar, that Black mothers have significantly higher rates of cesarean deliveries compared to other mothers. So why is that? Well, you know, and let me walk you through this story. So we know that Black mothers have higher rates of adverse outcomes. So what we could think, you know, we could really use AI to predict risk of adverse birth outcomes to better identify these risks and better align additional resources. So we want to address the differences to best serve diverse populations. And so we might want to include race in these models. But it's important to note that race is a social construct. And when we use race in models, we really need to justify why we're using race in these models. Is it really race that's associated with our adverse outcome, or is it one of these social constructs? So we've been looking at the vaginal birth after cesarean calculator, the VBAC calculator. The VBAC calculator was developed in 2007 by the Maternal Fetal Medicine Units Network. And here is, I'm sorry, it might be a little blurry, but I hope you can see that. Here is what the VBAC calculator included. It included maternal age, height, weight, body mass, African American as a binary feature, yes, no. Hispanic as a binary feature, yes, no. Previous vaginal delivery, at time, etc. And so we included race and ethnicity as a predictor of successful VBAC. Well, that meant that what that meant was that Black and Hispanic women were given a systematic correction factor for the odds of having a successful vaginal birth after a cesarean compared to white women. This led to disparities. We led to higher cesarean rates among Black and Hispanic women in the US compared to whites. And they were also offered less treatment options, right? Because they were identified as having a higher risk of adverse outcomes. So the calculator encoded racial differences, reinforcing inequities rather than addressing the root causes. So, you know, fast forward, the Maternal and Fetal Child Network started looking into this. Why are they including race as a predictor? And so what they did was in 2021, they launched an investigation to really understand the relationship between race and birth outcomes. And what they found was that race was actually a proxy for chronic hypertension. But we know that anybody can have chronic hypertension. It's not just associated with Black mothers or Hispanic mothers. So by removing race from the calculator, and actually including chronic hypertension, the calculator improved for everyone, not only Blacks and Hispanics. So I think this is really important when we start thinking about, you know, when we think about health equity and really understanding the association between some of these vulnerable populations and outcomes. It is really about improving the outcomes for everyone. And so we've taken this a little further and we've really thought about this across several different populations. And so our work really centers on vulnerable populations in healthcare and how do we ensure that when we're asking a question, these vulnerable populations are specifically accounted for. Right. So in addition to the VBAC calculator, we've also been doing some work looking at the use of telemedicine in urban versus rural communities. We've been looking at medvac transfers for helicoptering into emergency departments among Alaska Native communities. We've been looking at how depression and patients who are diagnosed with depression really have different likelihoods of treatment adherence. And how do we account for those specific populations, their specific needs when we're trying to predict treatment adherence? We're doing some work in elders and how a lot of the algorithms that are being used on elders do not actually contain this elder population in their training data. And so you can imagine that these differences are quite large. In addition, we've been looking at patients with disabilities and pressure ulcers. Most of the algorithms exclude patients in wheelchairs from pressure ulcers who are at really higher risk of pressure ulcers. And also we do a lot of work in pain management and opioids. And so really understanding patients with chronic pain, patients with type 1 diabetes actually have different needs for pain management. Yet a lot of these policies and algorithms are very generalized, are very general, right? They're for treating the average patient. But we know there are no average patients. And so in our work, we really think about how do we think about these vulnerable populations to ensure that the tools we develop are inclusive. Next, I want to talk about AI deployment and utility. And here's a big gap in our knowledge and in the literature of how do we move an AI tool that's developed in research into the clinic. And let me give you a key example. So and let's talk about sepsis. So this is a good example of why these models fail. So Epic, which is our electronic healthcare system, developed an early sepsis model several years ago. I think it was prior to COVID. So the work involved over 77,000 hospitalizations, and they had a sepsis rate of around 5%. So when the model was developed, it had a performance of about 80%. So they were about 80% correct in identifying patients with early sepsis. However, when that model was deployed, that accuracy dropped down to 0.62. And then under further investigation, when the clinicians started looking at that, they noticed that included in the model were tests that you would only order if you suspected sepsis. So once those were reduced, the actual prediction of the algorithm fell down to 0.047. So you would have a better chance flipping a coin to guess who was at high risk of early sepsis than using this model. So this is a key example of a very large model that was tested across hospitalizations that actually failed, right? And it failed because it lacked the correct variables. It lacked the diverse stakeholders to understand how and where it would be implemented. So we've been thinking about a framework for how we safely move these tools into clinic. So this is some work that we just did that came out. Sorry, I'm not, there we go. That came out, just a couple of weeks ago. And this was some work we did with Brigham and here at Stanford. And we're looking at how do we think about deploying AI into clinical care? And we took a randomized controlled trial framework. This means that first, before we even think about moving the model to clinical care, we think about the safety of the model. This is retrospective, right? We look at the model performance. We engage diverse stakeholders to ensure the questions we're asking are applicable to the populations to which it would be applied. We think about development strategies to prioritize underserved populations and really go through all of these analyses in addition to performance to understand the fairness of the model and the applicability. The next phase is phase 2, which is controlled implementation. This is about the effectiveness, testing the model in real time. We also call this the silent control, right? This is where we work specifically with one clinic and we run our model on that clinic, but we don't allow the clinicians to act upon our predictions. So here we're really assessing the effectiveness and the fairness of our model. We're ensuring we can get the data at point of care in real time, and we're ensuring that our model performs the same in real time as it did in our retrospective phase. The third phase is broad dissemination, the efficacy of our model on health outcomes. Does our model actually improve care? Does it improve patient outcomes? Does it improve something in the healthcare system? And a lot of times we're so excited about the hype of AI that we're rolling these out without really thinking about the impact on the system, on the patient, and on the clinician. So in phase 3, we expand our deployment across multiple settings. We compare outcomes of the AI tool between standard of care. Is there a difference? Is it an important difference? Is it worth the cost of implementation? Is there an increase in clinical burden? And it's important in this phase to maintain transparency with stakeholders about the performance metrics. The fourth phase is post-deployment evaluation. And here's where we have a huge gap in what we know about AI deployment, because this is really assessing the societal impact of the tools, assessing long-term effectiveness and equity. This is about really publishing findings and insights about how the tool has performed in communities, how the goals of the tool and the goals of your AI model align with community objectives. And establishing feedback loops, right? If things don't align, if the clinician is rejecting your tool, dismissing these triggers continuously, how do we incorporate that feedback into the tool to improve it, to make it better? And in this post-deployment phase, it's also about real-time monitoring. And this is really challenging, right? Because a lot of times we think we spend all this time developing the model. We spend a lot of time thinking about how do we deploy it, and then we're done. But no, we're not done, right? Because we need to continually evaluate the model. We need to continually ensure that the model performs in real time the way we expect it to. This means really understanding how concepts change over time and how data shifts over time, right? Let's say we deploy the model at a hospital, and the hospital gets a new insurance company, gets a shift in their population. How do we ensure our model's still going to work across these shifts in patient demographics? There might be a new drug on the market. How does our model perform once this drug starts improving patient outcomes? So all of these are key factors when we think about model deployment. And none of this is possible without community-engaged AI. And I think this is really important. And this is some work that Brian Rice has done in my lab, where we're working with Alaska Natives to understand how we can develop AI tools that are applicable to their community and meet their needs. How do we ensure that when we talk about community engagement, it's not tokenism, but it's really actively listening to their needs and what they want to get out of the project? So in this partnership with our Native American, Native Indian, and Native Alaskan communities, we're really trying to address the historical research abuses that have happened to these communities. We're trying to ensure that the data is safe and the privacy issues are preserved. And it was interesting because in this work, we first came, okay, we're going to bring, some people from your community to Stanford to really train them so that they can understand the technologies and the tools we're developing, so they can go back to your community to really help deploy these tools. And we got big pushback by that because we didn't really understand the community. And what the Alaska Native said was, it's very difficult when you bring our community to Stanford and then having, a lot of times those people do not return back to the community. They stay at Stanford, they stay in Silicon Valley, they find these other opportunities, and they don't go back to the community. So instead, they said, we want you guys to come to us. We want you to come hold your workshops here in our communities. Yes, Stanford might be a very rich environment where we can learn so much, but we want to preserve our community. So actively listening to these conversations, actually incorporating this information into our projects and how we build our tools is essential. It took us 3 years to actually get this project off the ground. And that 3 years was about building trust within the community, within the stakeholders, and really actively listening and engaging. So I think that's a key point when we think about inclusive AI. And so the last point I want to talk about is policy and standards for medical AI. And right now, I usually have a slide about the wild, wild west, because it really is the wild, wild west. There are limited policies and standards for controlling, or controlling is not the right word, for understanding and regulating AI in the healthcare system. So my group's been thinking about this for a long time. And how do we move forward with standards to really help inform policy? And first, we have published some work on reporting guidelines, right? When we're reporting our AI tools, what is the minimal information that needs to be reported to ensure the responsible use of the tool after the reuse of the tool? And in these guidelines, we said, you know, you really need to know details about the population from which the data were acquired, the training data demographics, the model design and development, as well as the model evaluation, optimization, and validation. If you have these key aspects about the design of the model, you can understand better if it's applicable to your population. We work a lot with community health settings, safety net hospitals, and it's really important for them to know, if I develop a tool at Stanford, how close is their population to the population from which we trained it on? What variables are we using? Why did we choose model A versus model B? And this is really about transparency and explainability. And this is an article we just had come out 2 weeks ago. This was a workshop that I co-chaired at the National Institutes of Health in 2024 on transparency and explainability for AI in health. And we convened a group of experts to really think about, what does NIH need to think about when we're talking about transparency and explainability? And it was a lot about data provenance. We're seeing more and more tools that are using synthetic data. So how do we track that data? How do we understand about data provenance? We thought there were opportunities to expand IRB capabilities to cover AI-specific ethics. We said we really need more training programs on AI's ethical, societal, and transparency needs. We're rolling out these tools so quickly that oftentimes the clinicians are not trained in how to interpret the outcomes. They're not trained in how to think about how these tools might be biased, or how the fairness matters. Trainees and end users often don't know how to interpret these data. We need to establish incentives for comprehensive documentation. We've done a lot of work thinking about data sheets and model cards and how important those are when we're describing the AI tools we develop. But what are the incentives for industry, for academics, for nonprofits to really take the time to fill out these documentations? There's not a lot of incentives. And so there's a big role here where policy can really help incentivize documentation, right? Collaboration across disciplines to enrich AI governance and address ethical needs is really important. When we're building our tools in silos, we can't understand the impacts or the questions we're asking and how applicable they are to other populations. In this workshop, we also talked about creating incident databases, or maybe proxy variable registries, to understand the risks. The FDA has an incident database for devices, right? And so they track when a device harms an individual. Well, when we think about AI, it's not about harming an individual. It's about harming populations. When AI doesn't work for our Black Americans, as we saw in the VBAC calculator, our Black women, it's not one individual that's harmed, it's the entire population. So how do we track that? And if we had an incident registry database, we could really understand where things can be improved, where we need to think about diversifying our training data, and where we need to think about where we're using these proxy variables and why. We've been working with the WHO to think about a global framework for ethical AI, right? And this is with the GI AI 4H. And we spearhead novel on-the-ground efforts, right? We're working with WHO to think about how do we think about these low to middle income countries and how they need AI, how they can benefit probably the most from AI, but how do we ensure they're ethical across these different populations? So in this work that came out earlier this year, we set up a global framework for thinking about ethical AI. This really was about capacity building, right? How do we think about developing training courses and tools for ethical standards that are globally applicable? We thought about how do we identify regional consultations for local adaptation. It's really boots on the ground. How do we have these communities understand the right tools that they should be implementing or they can implement and prioritize the tools? We also said there was a need for regulatory harmonization, right? There's a lot of fragmented health policies globally. I give a course on responsible AI. And right now in our course, we're talking about these different policies, global policies across the world, right? In the US, we really prioritize risk management. In the EU, they prioritize individual rights. In China, they're prioritizing innovation and fast moving forward. So all of these policies are fragmented. So how do we bring these together in a global framework and really strengthen international cooperation? The third aspect of this framework was equity by design. We want to be sure that the questions and the tools we're developing are really being developed to promote health and healthcare and well-being of the patient, of the individual. We want these tools to be centered on equity, inclusion, and human rights. One of the things that was really important in this work was thinking about large language models for healthcare. Sorry, I have a typo there. Large language models for healthcare, international healthcare systems, how they might benefit from these, but what are the guardrails we need in place? We know that these large language models, the ones coming out of industry, are trained on really diverse data. And in fact, with the latest generations of these tools, we don't even know what they're trained on. So how do we ensure there's guardrails in place so that these other nations and countries can use them? And then it's also about sustainable implementation. We don't want to just go to these countries and develop a tool and then walk away. We need to think about how do we strengthen the health system infrastructure to ensure these are sustainable opportunities, establish stakeholder accountability mechanisms. So to wrap up this morning, I think there's this real balance between promise and peril, right? I talked about some of our biological work we're doing with genomic data, right? So this is sort of a biological crossroads where we're at this point where innovation is just skyrocketing, right? We have so many opportunities to innovate and to improve the health and the well-being of individuals, accelerating drug discovery, precision solutions, genomic medicine. But on the other side, we need to be very wary about the adverse effects of some of these tools. In our biological work, we're looking at dual-use pathogen engineering, amplifying existing biases, and equity washing without inclusion. What does that mean? That means we're really just looking at the majority population, and these minority populations, these vulnerable populations are often excluded or not prioritized. So yes, the tools that we're developing, that we're designing, can design new malaria vaccines or drought-resistant crops. But that same model could also deepen health disparities if trained on unrepresentative genomic data. And so there is this key need to think about governance for AI and security. In our genomic work, we put out a paper thinking about this, really highlighting this call for governance, mandating pre-released evaluations of advanced biological models. Government should require independent biosecurity evaluations. We first need to, these mandatory evaluations are really important when we're developing these high-risk models. We need, again, as I said, international harmonization of oversight standards, just like the work we're doing with the WHO and thinking about these global incentives. It's similar for our genomic models and our synthetic data models. And so to wrap up, I think that there are, in what I hope to display, is that there's 3 pillars of equitable, inclusive AI. It's about the data and how we think about that data. It's about taking a research model and deploying it responsibly into either the healthcare system, patient-facing products, or to the clinical decision support. And it's also about policy, or the lack of policy. How can we really move policy forward? It's by developing evidence. It's about developing evidence to show how these tools can be used across communities and where harm might be highlighted. So I think that my research in my lab has really kind of given this roadmap for how we think of all these things, from community co-design to data justice. We think about algorithmic repair, mitigating bias, real-world validation, and really policy enforcement or policy-driven evidence. So to wrap things up, this is a paper we took out we wrote in 2023, again with my colleagues at NIH, about responsible AI and what are the guiding principles for in health and healthcare. And we said there's really 5 guiding principles for responsible AI. The first is designing questions and algorithms to promote fairness and reduce health disparities. The second is to ensure the questions we're asking are inclusive and representative. The third is engaging diverse stakeholders, community members, to ensure that we're actively listening to the societal impact of the tools we're developing. We said it's essential that any tool that's coming out needs to assess fairness issues and that trade-off between accuracy and fairness. And we need to think about accountability. When things go wrong, who's responsible? Is it the developer? Is it the healthcare system? Is it the clinician that's actually using the tool? Who's accountable? And how do we start thinking about that conversation? So I will stop there. Thank you so much for your time today and the opportunity to share our work, and happy to take any questions.
Thank you, Tina, for such an engaging and interesting presentation. I really appreciate it. Do we have any questions in the Q&A box? All right, there is one. How do you evaluate the real-world impact of AI-driven clinical decision support, especially when it comes to underserved and historically marginalized populations?
Yeah, that's a great question. And, it goes back to community engagement, right? We have to really look in the communities for how these tools are impacting different populations. And this is a question of trust, right? Because a lot of these underrepresented communities have been harmed in the past by AI tools. And so we really have to prove to them that, number one, we're listening to their needs and their concerns. And number two, that we're trying to develop tools that are more inclusive and really addressing their needs. But it is about building trust with those communities. And so, for example, in the work we've done with the Alaskan Natives, as I said, it's taken us 3 years to build that community. But now we're really excited because everybody wants to be involved. People are giving us feedback. And so it's about actively engaging with them and taking their feedback and really doing something with that feedback they give us. And so it takes a long time, but it's about engaging these diverse stakeholders. I think that's a key point.
That's very good. It kind of reminds me of like just getting your foot in the door. And then once you establish trust and a good working relationship with a group of people, does it help? Does it travel word of mouth? You know, it's easier to move from groups to groups.
Yes, absolutely.
Very good. Thank you. There's another question. Can you speak a little bit about the interdisciplinary collaboration in your lab? How the computer scientists, the clinicians, the equity researchers, how do they all work together to develop?
That's a great question. And I'm so proud of our lab because I think it is very diverse across every aspect of diversity. So what does that mean? We have high school students all the way to mid-level faculty involved in our group. We have people from health policy, from of course, clinical care, epidemiology, statistics, bioengineering. And so very, very diverse team. And so what that means is that when people come to us with questions, because all of my questions, any type of clinical question we answer, we're always partnered with somebody, a clinician or somebody knowledgeable on that particular clinical question. Anytime anybody comes to us with those questions, we come at it with diverse perspectives. Let me give you an example. There's an example of the hospital had a clinic that had high rates of no-shows in the clinic, right? And so we can very easily build an algorithm to predict who's going to be a no-show based on past information. But taking that to our diverse team, they're like, well, why would we be developing an algorithm to identify no-shows instead of thinking about what are barriers to access to care? Can we identify which patients might need transportation? Can we identify which patients might need a particular time slot for their clinic because they have caregiving obligations? And so really thinking from these diverse perspectives, we can flip these questions to ensure, going back to the guiding principles, we're asking and answering questions that promote health and health equity.
Very good. Thank you. I have 3 more questions. We'll see how many we can get to in 5 minutes. One of the hidden assumptions in AI systems is that the distribution is Gaussian, whereas you may have fat tails. An example is when COVID hit. Have you found instances where the actual distribution doesn't match the AI?
Well, that's a great question. And we talk a lot about data distributions and how we think about that, right? And COVID is a great example. Because all of our assumptions about concepts, about relationships between our variables and our outcomes went out the door, right? And so I always say, and in fact, with my students, you really have to understand your data distributions from your training data to where you're actually deploying the model. And this is the example I gave. Our populations are constantly changing. We're adding new insurance companies. We're adding new treatments, new patient groups. And so understanding these distributions is key when we compare our training versus our test set. And COVID is a great example. It's the example we're all using to think about that question.
Very good, thank you. And then one last question. How can you continue focusing on inclusion and equity of diverse communities when the current federal administration is dismantling it all and unfunding DEI efforts?
Yeah, that's a great question and something question I've been getting a lot recently. And so, I think it's important that we stay true to our values. It's really important that when we're thinking about equity and diversity and inclusion, what I say is, what is the fundamental basis we're trying to do, right? We're trying to think about these underrepresented populations. So I can come at it from a mathematical perspective, right? How do I think about ensuring that the algorithms we're developing are inclusive or applicable to these populations? And so what I showed in the work today is, yes, we look at race and ethnicity, but we also look at other types of vulnerable populations, such as populations with mental health. We look at populations with diabetes. We look at populations with disabilities. And so I think there's still opportunity to understand health equity, but I think we need to, as in any field, right, we need to really ensure that when we're speaking to different audiences, we're tailoring our conversation to really be able to move forward with what we care about. And so people have come to me and said, do I stop looking at health equity? I said, no, absolutely not. But we need to think about what is the question we're trying to solve, right? We're trying in our AI world, we're trying to solve the question about generalizability, about ensuring applicability. And so let's think about addressing those questions and maybe toning the conversation to the particular audience that we have right now. So hopefully that could answer that question.
Yes, definitely. Thank you. I think we are right at 10 o'clock, so we are on time. I would like to thank you for your time to share your expertise and your thoughts and insights. And thank you so much.
Thank you
for being here today. Thank you. All right, so we are right on time. It's 10 o'clock. We can transition over to our data-driven medicine, women in health informatics panel discussion. Today we have three wonderful panelists. I will go ahead and introduce them one by one, and they will have a few minutes to talk about their research in health informatics. Then I will facilitate a panel discussion. We do have some pre-prepared questions, but to get the conversation going. But as that's happening, please make sure that you put your questions into the Q&A box and upvote them so we can get to them if time allows. And with that, I would like to introduce our first panelist, Dr. Sandhya Subramanian. Dr. Subramanian is an assistant professor in the UCSF-UC Berkeley Joint Department of Computational Precision Health. Her research focuses on developing innovative, non-invasive wearable technologies, along with algorithms and computational models to better monitor patients in at-home settings and provide valuable information for data-driven clinical decision-making. She is also affiliated with the Berkeley AI Research Lab and the UCSF Department of Neurosurgery. Dr. Subramanian completed her undergraduate studies in biomedical engineering and applied mathematics and statistics at Johns Hopkins University in 2015, followed by a Master of Philosophy in Clinical Neurosciences at the University of Cambridge. She completed her PhD in medical engineering and medical physics at the Harvard-MIT Division of Health Sciences and Technology in 2021. Her thesis work involved building interpretable models to track unconscious pain processing during surgery. Most recently, she was a Schmidt Science Fellow and Stanford Data Science Postdoc Fellow at Stanford University, where she developed systems and inference methods for continuous at-home physiological monitoring for complex chronic diseases such as migraine and functional digestive disorders. She was named a L'Oreal USA 2022 for Women in Science awardee and a Rising Star in Engineering in Health. Welcome, Dr. Subramanian. I will turn it over to you.
Thank you. It's great to be here, and thank you for inviting me. I have a couple slides to help facilitate kind of sharing a little bit about what I work on. Okay. All right, can everyone see it in presentation mode?
Yes, we can.
Perfect. Okay, So the focus of what I do in my lab is thinking about how we monitor and model the body outside of the clinic. I started off a lot of my research in clinical settings, including even in my PhD, I was in the operating room. And that led me but the work that I was doing led me to using a lot of wearable sensors. And then that led me to thinking about how that then enables us to actually continue the same kind of rich data collection and inference that we can do inside the clinic, outside of the clinic. So the question that I spend a lot of time thinking about is why is information exchange in healthcare so dependent on, why is information transfer so dependent on doctor-patient exchange? And what I mean by that is the current healthcare system's main paradigm of how information is transferred is that a person comes into an appointment and then they tell the doctor a bunch of stuff about how they're feeling, what's happened since the last appointment, the highs, the lows, what they think needs fixing. And then the doctor takes all that information in and makes a decision, maybe adjusts their treatment, maybe suggests something new, maybe refers them to a different specialist, right? But in reality, if we look at all of our lives, we spend a very small fraction of time in a clinical setting. And we spend the majority of our time living life outside of the clinic. So that includes our diet, our sleep habits, our physical activity, our social activity and interactions. And all of these have direct effects on our health, but they are largely out of the purview of the healthcare system unless we choose to bring it up to a clinician at an appointment. So what does that mean? It leaves out a lot of valuable information. So everything that happens outside of the clinic. There are a lot of situations in which patients cannot be good advocates for themselves. That could be because of a health condition that they have that alters their ability to keep track of or perceive information about themselves. It could be because they live alone, and so there's no one else who's living with them that is able to observe things about them. It could be because of language or cultural barriers that makes it hard for them to communicate with the clinician in an appointment. It could be just logistically because it's difficult for them to make time to come to appointments often in order to relay information. And then there's an entire layer of information that is complementary to and outside of what any patient, even the most observant patient, could perceive about themselves because it's below the level of their conscious control. So I kind of think a lot about these categories of information. So how do we enrich what the healthcare system has access to by giving, kind of shedding light on these situations? So what that ends up meaning is that I spent a lot of time thinking, from a physiological standpoint about the system in the body called the autonomic nervous system, which is the branch of the nervous system that controls everything that you don't consciously think about, right? So I have a bunch of examples on that figure there. That includes our heart beating, breathing, digesting, food, kidney function, hormone production, reflexes that we slightly adjust our posture and it changes to make sure our heart rate and our brain is still getting blood flow. All these small things that are happening automatically throughout your body and you pay no attention to. Now, what's great about the autonomic nervous system is it hits all of these three marks, right? So it's always functioning, whether you're in a clinic, outside of a clinic. It doesn't matter whether you can communicate. It doesn't matter whether you're unconscious. It doesn't matter whether you're asleep. It's always functioning. And by definition, it is below the level of your conscious perception. So you could not, even if you wanted to, communicate about these things to your clinician. In terms of opportunities that I think about and my lab spends a lot of time working on, I am myself from a very interdisciplinary background. So you might have heard in my introduction, I'm trained as an engineer, as a biomedical engineer, also as an applied mathematician and statistician, and also as kind of a neuroscientist, and I do my own clinical studies. And even though I don't have a direct clinical background, my PhD involved a lot of clinical training. And so the first question I like to think about is how do we engineer wearable sensor systems for the real world? So that includes prototyping new hardware, building new sensors, thinking about where there's opportunities to collect new information. What new models, machine learning and AI models, do we need to build to understand signals from the body? So this is kind of the computational domain. And then finally, where are there untapped opportunities in clinical medicine to better understand individual physiology? So new application spaces. I think there's a lot of untapped potential for wearable monitoring. So first, scientifically, it allows us to capture individual variability and complexity in physiology in a way that we cannot in limited snapshots in a clinic. It also allows us to track the effects or the outcomes related to personalized treatment strategies. So during my PhD, as in the intro was alluded to, the work that I did was around tracking unconscious pain when someone was under anesthesia. So they're unconscious, so you can't ask them how they're feeling, but their fight or flight reflex is still intact. And so you can use wearable sensors to track aspects of their fight or flight response, and then develop a computational model that tries to predict when their body is still processing unconscious pain. So this is for a single surgery, for example, and on top I have these kind of cartoony symbols of like when there was different kinds of painful stimuli administered as part of the surgery. So whether that be placing the breathing tube, making incisions in the skin, or cauterizing or burning internal tissue. And then the model is able to kind of follow along with their physiological response and give a prediction for when the body seems to be processing a high amount of this noxious stimulus. And this was published in PNAS last fall. Another kind of really important upside of wearable monitoring is that it's very scalable. It's not, so everything that I do is non-invasive. It can be done 24/7. I often have patients wearing sensors overnight, and it can be at home. So I send them home with them. So they can sleep in their own beds and live their own lives and eat what they would normally eat. So this is an example. These are all examples of different prototypes or different sensor systems that we use in the lab. And then obviously, the end goal for everything is impact. So we want to provide the clinician with richer insight, and we want to maximize the patient's quality of life. So this is work from my postdoc at Stanford, where we had 24-hour at-home data collection from patients who had a functional digestive disorder called gastroparesis, meaning that food sits around in their stomach for a lot longer than it should. It doesn't move to their small intestine. And it leads to a lot of unpleasant side effects because your body does not like that. And then we also had healthy people as contrast. Now, even if I told you nothing about what these axes represented, right? If you just looked at squiggly lines for each person and you saw when they were awake and when they were asleep, there's a difference between the two people. So the healthy person, there's a clear separation between wake and sleep. Something changes. All the squiggly lines look like they're doing something different when the person's asleep than when they're awake. Versus in the patient who has gastroparesis, a specific subtype, diabetic gastroparesis, so they get it as kind of a ongoing sequelae related to their diabetes. Nothing changes between wake and sleep. It's basically indistinguishable. Now, all of these different measures are actually related to the autonomic nervous system, and specifically they're related to control of the heart over the course of the circadian rhythm, which seems a little not intuitive because this person has a digestive disorder, right? But the interesting thing about the autonomic nervous system is it has this very interesting property of turning local problems into global problems, right? Like turning local problems into a more global stress response or stress manifestation. The other thing that we were able to do, and all of this was published in this paper from 2 years ago, is we were able to track the same person over time. So we did 24-hour snapshots of this person before they had a surgery to fix an obstruction at the end of their stomach, a month after surgery and a year after surgery. And what was interesting is that, again, don't have to pay too much attention to what the axes mean. I just told you the answer key, which is moving up and to the right is more like a healthy person. A year after surgery, they look much more like a healthy person in terms of their overall circadian governance and their overall circadian regulation of even their heart, even though the thing that was fixed is a very localized thing in their digestive system. So wearable monitoring gives us the ability to have this level of nuance and precision even on an individual person scale. And then we were able to turn this into kind of prototypes of diagnostic scores that could help differentiate different subtypes of disease. A lot of what I do is building what I think of as physiology-informed AI. So how do we marry the power of AI with the many decades and centuries of underlying physiology that we have understood about the body? So I use a variety of different methods. And I think of impact on a continuum. So it can go from, in diseases where we don't understand what's happening very much, we can go from anything from understanding about the disease better, right? Providing more insight about what's actually happening in the disease, to then building clinical decision support tools to enable clinicians to make better use of that information. And then eventually the holy grail in every case is have some sort of personalized prediction or closed loop control on an individual level. And I'll stop there and thank my lab, my collaborators, and please reach out if any of this sounded interesting or if you have any questions. And I have a snippet of the kind of ongoing work we have in the lab.
Wonderful. Thank you so much, Cynthia. I greatly appreciate you sharing this with us. And then at this time, I would like to introduce our second panelist. Her name is Luna Balbaki. Luna is a research data analyst at Stanford University's Center of Biomedical Informatics Research. She has a strong foundation in data analysis, machine learning, and statistical modeling. With a deep commitment to data integrity, Luna ensures that impactful science begins with high quality and well-structured data. She holds 2 master's degrees, one in economic development from the London School of Economics and another in business analytics from the American University of Beirut. Luna's career spans academia, fintech, and nonprofit organizations, where she has worked on impactful projects. In her various roles, she focused on building trustworthy data pipelines, ensuring the accuracy, consistency, and readiness of data for meaningful analysis. She is also passionate about using data to solve complex challenges. As today's speaker, she is excited to highlight the often overlooked role of clean data as the foundation of ethical and reliable research. Please welcome Luna, and I will turn it over to you.
Thank you very much, Boglarka, and good morning, everyone. And yeah, thank you for the introduction. It's an honor for me today to be joining the panel at the Women in Data Science Conference. And I'll start by sharing my screen. Okay, can you see my screen?
Yes, we can.
Okay, great. So today I'll be walking through some key dimensions that we can see for the data quality, and in order to check the critical role of data quality in health informatics. And also I'll be walking through some steps that we can take in order to enhance the data quality. And it's very important, especially in the medical field, because data quality and assessment can really affect people's lives and the decisions that we take. So to start, why data quality drivers matter in health? It matters because for research and innovation, for patient safety and clinical decision support, and as well for regulatory compliance and reporting. And it's important because every piece of data matter. So if we have a typo in lab results or missing blood pressure, can affect our analysis. It can affect the treatment. It can delay treatment. So that's why having good data really matter. And even our smartest algorithms won't give us the right prediction if we don't have if our data isn't clean and isn't well. And that's why it's very important for us to have accurate and validated data so that we can come up with the right decision making. And as well with the saying, garbage in is garbage out. So that's why it's very important for us to have good data. And here are some key dimensions of data quality. So some attributes of the data quality, one of them is accuracy. It's really important for our data to be accurate, because if like the blood pressure reading, how would we know that the blood pressure reading 120/80 is correct and not someone like added an extra 0? So that's why it's quite important. And also for completeness. So we need to make sure that we don't have any missing key fields, and we got all of the key fields that we need. Consistency. So make sure that all of our formats are consistent across the different lab systems that we have. And timeliness. So making sure that our data is up to date, we don't have any delayed data. And also validity. So this makes sure that our data meets the defined formats and standard, and all of the data that we have is within the plausible medical range. So we make sure that we don't have age as a negative number or blood pressure as a negative number. So all of these qualities are dimensions of the data quality. And we also have some challenges in the health informatics regarding the data quality. So one of them that we also tend to face is manual data entry errors. So typos, misplaced decimals can affect our data quality. So heterogeneous systems and standards, and that's when we have different softwares in our lab that don't have the same background or same standard software. And also when we have missing or delayed data, so when patients are seen offsite, that can cause some missing or delayed data, and also some semantic variability. And by that I mean when we have same term coded differently. And in terms of that, we do have some couple of procedures that we need to take in order to ensure that we don't have these errors in data quality and to overcome these challenges. And so for the best practice, what we usually do is that at first, we need to define the requirements. So we need to make sure what are the critical fields that we need. So we have the patient ID, lab code, timestamp, and we need to make sure that they are there for every patient. And after that, we do the assess and analyze. So we do some initial analysis and make sure that we don't have any missing fields or duplicate counts, because we don't want to have a record for a patient that doesn't have an ID or the patient name or important information. So we make sure we don't have missing values, and as well same for duplicate counts. And then the third step is we validate incoming feeds. So we have some schema checks, and by that I mean that we make sure that we reject records with wrong data type. So if a user mistakenly put a text for the age field, we make sure that we don't accept that field and we don't accept that value, because it should be an integer. And same with other fields that we have. So that in order to make sure that we do some validation to the data and we don't end up with bad data and unclean data. And also an important step is that we document, which is the fifth step. So we make sure that we always have documentations, wikis of the steps that we're taking, of the data that we have, in order to make it easier for our team members when they want to work on the same project. And the sixth thing, which is the control and report. So for here, after we do all of these steps, we make sure we have some KPIs to check the percentage of completeness, processing time, and also we do have some alerts when any KPI dips down below threshold. So these are the usual data quality processes and steps that we take in order to ensure that our data is clean. And finally, so for the data quality is important because for both business and the clinical impact. So for return on investment, when we have clean data, means faster analysis, means a shorter time to insight, which means cost saving. And all of these are important in the medical field. And also it's important for patient outcomes. So better predictive models would lead to earlier interventions. So when we have clean data, that would cause our models to predict better. And that would basically mean earlier intervention, so earlier treatment, which can basically help save lives. And also some collaboration with research. So high quality shared data sets fuel multidisciplinary research. So for all of these reasons, it's very important for us to make sure before we do any AI model or machine learning model to make sure that our data is clean, because often this is disregarded and seen as not important, but it's very important because so that we would have accurate and good results at the end. And in the healthcare field, data isn't just bits and bytes, but it's people's lives. So that's why it's always important for us to make sure of the data quality that we have. And thank you.
Thank you, Luna. Thank you. I appreciate that. That was very interesting. Learned some about, clean data and the importance and how we get to it. And then At this point, I would like to introduce our third panelist, Dr. Holly Tabor. Dr. Tabor is the director of the Stanford Center for Biomedical Ethics. She is professor of medicine at Stanford University and by courtesy of pediatrics and epidemiology and population health. She is also co-chair of the ethics committee at Stanford Hospital and the Lucille Packard Children's Hospital. She is a globally recognized expert on the ethical issues surrounding healthcare and research for patients with disabilities, especially intellectual and developmental disability, and on the ethical, legal, and social issues in genetics and neuroscience. She is editor-in-chief of the American Journal of Bioethical Empirical Research. Please help me welcome Dr. Holly Tabor.
Thank you. And thank you very much for the invitation. Can you hear me okay and see my slides?
Yes.
Great.
Yes, we can hear you and see the slides.
I'm honored to be a part of this and with such interesting and distinguished speakers. I really appreciate the opportunity to do this. I'm going to go through a few snapshots of some of our work thinking about disability, equity, and data science and health informatics. And I think it does connect to some of the themes from the previous speakers as well. I'd like to acknowledge with their permission the fact that part of my introduction to thinking about disability and health is by being the parent of two young men, Jasper 20 on the right and Colin on the left, and Colin 23 on the right, both of whom have disabilities. Through them, I am on a continuous learning curve that never stops, trying to learn and understand the perspectives of people with disabilities. And through them, I've gotten to meet amazing other people with disabilities, their family members and friends and caregivers and policymakers who work on these issues. So I just like to acknowledge that personal connection. This is a challenging time to be thinking about disability and equity in healthcare. This is just a few headlines. There are more from this week, sadly, but it's I've talked about this a couple of times now. This is, I believe, the single hardest time to be a disabled person in the United States. Certainly in my lifetime, certainly I think since the 1960s and 70s. One could argue that one of the biggest challenges is that there are public services that many people with disabilities have come to rely on that are being eliminated and are going to have substantial impacts on the quality of life of people with disabilities, and I do believe will cause many people with disabilities to die prematurely. And so that is the urgency which frames a lot of the things that I'm going to be talking about today, sadly. So I have been working with a wonderful team of people on inclusive healthcare for the last 5 or 6 years. And inclusive healthcare is this idea that people with disabilities have a right to equitable access to healthcare and full participation in health programs and services. It's also the idea that providers, health systems, and society have moral, ethical, and legal obligations to make healthcare more accessible to full participation by people with all forms of disability. By some measures, the number of people who currently identify as disabled in our population is about 25%, but most of us will have some form of at least temporary disability sometime in our lives, and almost all of us will have someone in our lives who identifies as being disabled, perhaps over many years and decades. To do this, we need all sorts of change, transformative change on an individual level, a policy level, and a technological and societal level. Just a few quick facts to frame the discussion, because many people don't know this. This is data that's a few years old from the CDC, focusing on adults with disabilities age 18 to 44. Arguably, this is worse for older adults. 1 in 3 don't have a usual healthcare provider. 1 in 3 have an unmet healthcare need because of cost in the past year, and that was before some of these current proposed cuts. And 1 in 4 did not have a routine checkup in the past year. There are also disparities in terms of violence and injury for this population. People with disabilities have a significantly increased risk of unintentional injury, and disability is as great or greater a risk than age, sex, gender, race, or education. People with disabilities are 1.5 times more likely to be victims of non-fatal violent crimes and 2 times more likely to be victims of rape or sexual assault. So this is the context in which my team began our work and does our work, which is that we need empirical data about the experiences, needs, and challenges of disabled people in healthcare. And how do we get that? So we formed a project and a team called IDD Transform. You can Google it and you will find our website. And our broad mission is to improve health, healthcare, and health outcomes for adults with intellectual and developmental disabilities in a measurable way. and really to engage those adults themselves in every stage of the research. And we do that using tools of community-based participatory research, sometimes called CBPR. The key principles of CBPR are equity, focusing on community strengths, focusing on action and research, and doing co-learning with the people who are actually affected by disabilities themselves. And in this work, we now have many more partners, but we started in the beginning, and they're still our partners with Special Olympics Northern California and an organization in San Mateo County called Partners and Advocates for Remarkable Children and Adults, or PARCA, which provides services to this population. This is a picture. We do almost all of our work on Zoom. This is part of our planning group, including people with intellectual and developmental disabilities. This is during the pandemic when the federal government sent out the free COVID test kits, and you can see that some of our participants were holding them up to the screen as we were talking about it and designing our project. This is a picture of our website. We have a core advisory board of people with IDD, family members of people with IDD, healthcare workers, caregivers, and community organization leaders. And they're a wonderful group of people. I also recommend you go to this website. I'm going to mention it again at the end, but we have a series now of about 17 virtual health forums we've done over the last couple of years, focusing on issues in IDD and health. So our question that we got to work on with Amanda and others in the library that I want to talk about today is what research has been published about the experiences of people with IDD during the COVID pandemic? How was IDD defined by a range of different studies? What subtypes of IDD or diagnoses? What countries and healthcare contexts was the research done in? And what demographic characteristics or populations, and how well were those populations described? Were people with IDD involved in the design and execution of the project? Was it designed to accommodate their needs in the data collection? What were the results and what were the policy implications? And so we're working on this right now. Our inclusion criteria were that it must be specific to the IDD population. We didn't want it just to be on disabilities generally. It must be inspired by COVID. We defined inspired by COVID. We didn't just want a project that went to virtual because of COVID and wasn't really looking at questions related to COVID. And it must be empirical research, including case studies. There were a lot of opinion studies. We screened, with the help of our library partners, over 3,000 abstracts. Right now, we're in the middle of looking at the full text. And so far, we've included 900 studies, which is far more than we thought. Some notes from our preliminary analysis is that, perhaps not surprisingly, most of these studies come from the caregiver or the medical provider point of view and not from the perspective of people with intellectual and developmental disabilities themselves. That's not always wrong. There are certainly some people with IDD who may have disabilities that make it hard for them to participate in data collection, but it's a bit concerning because there are certainly important things that get missed, particularly in the spirit of thinking that people with disabilities have important perspectives to share, which I'll show you in a minute. And most studies on COVID impacts on IDD discuss the impact on mental health and behavior, and from the perspective of self-advocates or caregivers, not looking at other health issues. One of the things we've done from this work is know about some of the tools and resources that are missing in the population. I don't have time to share this, but I can put it in the chat in a minute. We worked with partners at EdTech to develop a video about how people with IDD can ask for accommodations in healthcare. And we're looking at some of the papers that we analyzed, looking at issues and barriers to access related to how disabilities make it harder for people to get healthcare. I want to mention one of our other initiatives is a group called IDD at Stanford, which is an attempt to sort of connect the sometimes siloed, diverse groups at Stanford that may be working with patients with intellectual and developmental disabilities, including in data science, and also people with IDD in the community, students, staff, and community organizations. And we have a brunch once a quarter. We have one this Friday that everyone is welcome to attend, even from the community, from 10 to 12 at the Disability Community Center. And at those brunches, we always have a person with IDD. We always have a person from the community who works with people with IDD, and we have someone from Stanford who does work with IDD. And we have already fostered and nurtured several new collaborations that have come out from those groups. We also have an email listserv, so if people are interested, you can sign up for that listserv on our website. So in the interest of time, I'll end with a call of action related to our work in data science. I think we must listen to disabled people, and we must look at data about disabled people. I think this is very related to Dr. Hernandez-Boussard's work and both of the other speakers as well, about some of the biases that happen when we base our data on data that may not include the perspectives of all populations, particularly underserved populations or populations that have trouble getting healthcare. This is an old cartoon about Nothing About Us Without Us, which is a message of the disability rights and disability disability justice movement. This idea, again, that, perhaps it's well-intentioned of people to speak for people with disabilities, but in fact, in this day and age, it's actually bad data if we don't actually collect data about and from people with disabilities themselves. I like to end many of my talks with this quote from my friend Paul Miller, who unfortunately passed away in 2010. Paul was a short person who had achondroplasia, and he also had a genetic cancer condition called Leigh-Fraumeni syndrome. He was very involved in the disability rights movement. There's a picture of him on the bottom with Judy Heumann after the ADA and several other leading activists after the ADA was passed. And when he was approaching the end of his life, he was interviewed, and he said, "When I was born, I know my parents were scared. They looked down at this little shriveled child and they wondered what my life would be like. Would I play ball? Would I have friends? Would I be able to do the things they dreamed that their son would do?" And I have played ball a bit. I've always had fantastic friends. I have had good jobs, and I have done a good job at them. Along the way, I have helped other people. I have a lovely wife and 2 beautiful daughters. I have had a marvelous life. And I just want my girls to know that everyone can and must live a marvelous life. And so I think our message is to help people with disabilities live a marvelous life as they define it, and to use our different ways of looking at data and data science to try to maximize the ability to collect data that can help achieve those goals. So with that, I'll stop, and I'm happy to take any questions.
Thank you, Dr. Tabor. That was eye-opening and very moving, especially that last slide. All right, so we can transition into our panel discussion. At this point, if you could, if my helpers, if you could please highlight all the 3 big speakers. And we do have some pre-prepared questions, and please, audience, please continue, put questions into the Q&A box and we will answer them. So my first question is, and I will ask the question to all of you, and then feel free to unmute yourself and respond if you feel like responding, or if you think this is something you would like to speak on. So the first question is, can you share a project or moment in your career that made you stop and say, this is why I'm doing this?
It's hard to choose one. I can try.
All right.
Am I going first? I think so. There's a data science component to this, but it's sort of downstream. I hope it's okay if it's not specifically data science linked. But one of our projects recently that I didn't have time to share the slides of is a collaboration with Dr. Abby King in epidemiology and population health, using a tool she's developed called Our Voice, which is a tablet phone-based app that allows citizen scientists to collect data about things in their environment and curate and analyze that data and make recommendations to achieve change. And we use that in a clinic setting with people with and without intellectual and developmental disability to look at how we could make clinic spaces more accessible to people with disabilities. We've done it twice now in 2 different clinic settings, and we're hoping to do it more. And I have to say, going through clinics with these teams with their tablets, taking pictures, they take a picture, they code it as positive or negative or both, and they write a comment for every picture. And then we have community meetings afterwards to analyze the data. And I am just so moved by the quality of the data and the perspectives that those people with disabilities take. And I think it was a moment for me of realizing the importance of centering patients' voices. And so that's just one example.
Thank you, Holly. Luna or Cynthia?
Yeah, sure. It's a very good question. And I remember, one time, researchers, one of the projects, the researchers came to us and told us that their ML model is not predicting well for elderly women, and they want us to check why is that. So we dig deep into the training data, and we did some analysis, and we found out that the data is biased and there is not enough data for elderly women in the training data, so they are not well represented. So what we did is we did some oversampling, some techniques in order to make sure that the data is balanced, and then we gave them the right models and everything. And after around couple of months, they came back to us and told us that now their model is predicting better for elderly women, which means that they can now start with early interventions. So that made us really happy at our like team, because we're seeing the impact of it.
Very good. Thank you.
I'll keep my answer short, but I think for me, a lot of these moments have happened around mentoring, which is another really fun aspect of doing science in academia. So I think a lot of my like moments of feeling validated in my choice of career have come when I have mentored people and seen the like their transformation into both like loving the work but also realizing the impact that they could have. Also when I get to take them into clinical settings and like actually talk to the patients as we're like putting sensors on them or whatever. That's another part of the experience that I think has really made it come full circle for mentees, which has then for me made it feel very rewarding.
Very good. Thank you. Next question is, so this is a Women in Data Science conference, emphasis on women, and all of our speakers are women. That's one of the requirements that we have. So this is relating to working as a woman in a very male-oriented field, historically male-oriented field. As a woman in data-intensive and the male-dominated fields, what strategies would you recommend to early career women looking to find their footing in these spaces?
I can go first if that's okay.
Sure.
Especially for early career women, I think a couple things were helpful to me, and I've seen helpful to other people more recently who are more early career in recent years. One is, well, a couple things. One is really cheesy, which is figuring out your why and remembering that it's a marathon, not a sprint. Because I think particularly in early career for anybody, but especially for women, there are a lot of signs and messages that can be discouraging. And so if you have a why, I think it can help motivate you to connecting to your why through what will inevitably be some frustrating moments and disappointments. I hope for a time when that's less the case for women, but I don't think we're quite there yet in a lot of ways. I think finding mentors, realizing that mentors are not always women also. Some of my best mentors who've been most encouraging of me, and particularly combining my work and my career path as it's taken different turns and combining that with parenting and parenting to children with disabilities, have been men. And I think that was a surprise to me. I thought women would be better mentors. I've definitely had good mentors too, women mentors too, but sometimes mentors come in unusual phenotypes. And so seeking out different mentors and realizing that no one's going to be a good mentor for everything. One of my mentors early on talked about a mentoring family, that you have different mentors for different kinds of things that you need. You maybe have a mentor who's tougher on you, and a mentor who's your cheerleader to you, and your mentor who's helping you with this thing, and a mentor is helping you with that thing. So I think thinking about developing a mentoring network that you can sort of nurture and sustain throughout your career. I think the other thing is realizing that tenacity counts for a lot. And, you're going to have years that feel frustrating for all sorts of reasons. And you're going to have a system that often doesn't encourage you when those frustrating things happen. And so realizing that, again, going back to my original point, it's a marathon, not a sprint. Sometimes just sticking with it and doing the next best thing and getting past a particular challenge, I think, is one of the most important things moving on. And then the last thing I'll say that I think about a lot, which is how do you connect to other women, both other women who are your cohort or your stage of career, but also people who are coming up behind you. I think that's really, really important, and being sort of open to more informal connections to the people. I have a lot of people come up to me after talks and email me and things like that, and I think that's really important. And then it's okay for you to be that person too. So yeah, that's a couple thoughts I have.
Thank you.
Yeah, I have as well some suggestions. So, especially for early career women, I think one of the important things is to have a portfolio or a log of all the projects that they work on. So maybe put all of the projects in a GitHub or on a website, because a lot of people later on tend to ask on for like a reference or like for their portfolio, so they can just like send the link, and they don't need to like look within their folders and find it. So I feel that I learned it the hard way. And also, as Holly mentioned, it's very important to build their network. And also to do so, they can join conferences, workshops, and there they can meet a lot of people from that work in the same field, but on different things. So I feel that's very important.
Thank you.
I don't think I have anything to add to both of those answers. So in the interest of time, I will say second to both of those answers. We can go to the next question.
All right. Thank you. The next question is around diversity. And many of you have, or most of you have touched on this in your short presentations. How do you believe that diversity in data science teams can impact the outcomes of healthcare projects? And if you have any specific examples.
Well, I can start and I'll be brief so that people know. There's no way our research wouldn't be the same and wouldn't be as rigorous and accurate without the inclusion of diverse people in our research team. So I see that every single day. I think Dr. Hernandez-Boussard also talked about this, that much of the data is based on white insured males that that is done for AI related research in particular. And so if we don't have diverse teams and we only look at data that's from a subset of the population, then our results are going to be not very helpful or not very helpful for the majority of the population or for policy development. So I think it's really important. I also think to the point that someone asked her about what we should do in this current environment, I think we have to continue to find ways to do it, to be inclusive in what we do. I'm thinking about that all the time and talking to people about that all the time, but I think that's important.
Very good. Thank you.
I'll also say on the other side of it, the people doing the research, also like the work I do is inherently interdisciplinary, so it does not function if you do not have people coming from very diverse backgrounds, and training coming together and bringing their various strengths. So engineers, data scientists, clinicians, neuroscientists, physiologists, ethicists, people who study ethics, right? So like if you don't have people from various different backgrounds coming together, that is, it's another type of diversity.
Thank you. Luna, did you want to say anything?
No, I totally agree. And I think on like the data science team itself, it's always important as well for the team to be diverse, because diverse teams tend to like spot different blind spots in data, and people like from different backgrounds and different education, they can always bring new and different solutions, which can lead to better results.
Very good. Thank you. I do have a question from the audience. This was earlier, and I think it was when Luna, you were doing your presentation. So I will start with you, and then Holly or Sandy, I can add as well. Is the data quality process automated or continual?
So we tend to make it automated, so that it would save us a lot of time. But we definitely need to do our manual checks or our manual edits and adding. So for example, for the automated process, if we find out that we need to add new validation criteria for a different like disease or model, we definitely do that manually. But for the whole process, it definitely is automated.
Very good. Thank you. I don't know if Holly or Cynthia has anything to add to that. I think that was specific to Luna's presentation. All right. I have the next question about training programs. If you could redesign a data science or biomedical research training program from the ground up, what key skills, technical, ethical, or interpersonal, would you ensure every graduate walks away with?
I can start. So I would say the one thing that I think I tell every student that comes my way as a general piece of advice is, even if your main goal is to be a data scientist or a computational person, learn how the data that you're working with was generated. Like on a very ground level, like go participate in the process at least once and learn where that data comes from. So it's not just numbers on a screen or words on a screen for you. You understand when data is missing, like what does that mean? What could have contributed to that? You understand the people who put in the work on the front lines who are collecting that data, like how much work they have to put into it and what their struggle feels like. When you see sources of noise or artifact in your data, it gives you way more perspective. So like every graduate student that goes through my lab participates in data collection. And I think that's a really important perspective to have as someone who deals with data a lot.
Yeah, and adding to that as well, for the technical side, I think it's important to teach students about like the data engineering process. So the whole process from data collection to implementation, because usually courses are either just a data collection course and then like ML and then visualization. But if we have a course that combines all of them and gives students practical examples for them to start with data collection and then finish with like an implementation on like a web app or application or something, I feel that's very important so that students can connect all the dots. And as well on the ethical side, I think I will definitely give students hands-on bias auditing exercises so that they can see, because usually in real life they're going to have data that is biased and the data is not going to be perfectly represented. So if they are trained with biased data, then they can think about solutions and what to do so that when they're in the real life they can know what to do with the data that they're dealing with.
Thank you.
I can say one thing. I just think from an ethics standpoint, all training programs need to think more about professional ethics in lots of different ways, but also ethics in society. I think that's sometimes the part of ethics that gets the least coverage in training programs. And so maybe that's even more important in data science. What are some of the obligations, including about communication about our research and results when we think about the role of researchers in society? So I think that's an opportunity.
Very good, thank you. I would also like to encourage the audience to put questions in the Q&A while we are answering my questions or the questions we predetermined. All right, there is an audience question. What is being done to address AI junk science research?
I'll answer really quickly and then I have to hop off early. I assume by that you mean like the now what is ubiquitously easy to do of the AI generated, research and other content that is not necessarily of high value. I will say, I think our ability to put checks on it is a little bit behind. Like, we're playing a little bit of catch up at every step of realizing like all the ways in which it can exist, and then we keep playing catch up, but we recognize that we can see it. So it's not just in AI generated research, it's also in any, like, we see it in applications, we see it in research proposals that people write as part of applications or in statements people write. We've seen, we've seen it this cycle a lot, and it like kind of exploded all of a sudden. So we're still playing catch up, but it is, especially when it is from the standpoint of trying to generate new scientific content, at least my experience has been, it is pretty easy to identify when you see it. It sets off a spidey sense pretty easily to figure out, like, I don't think this was this person's original scientific idea. But I think in terms of, systemic or regulatory trying to fix it, we're still catching up, we're still playing catch up.
Yes, thank you. Holly or Luna? Yes, thank you, Cynthia. I really appreciate your time and sharing your expertise and knowledge with us. I know you have to leave to catch a plane. All right. Another audience question. Do you have any free resources you can recommend to students interested in learning data science?
Yeah, definitely. There are a lot of courses available online and resources. I can share some with you over email if you want to share them with the attendees.
Sure, thank you. I will, yes, I will connect the two of you.
I don't think I have any specific resources, but I think there are increasing course opportunities for students that are good foundational things for this. And then I was just looking this morning, there are also some free conferences on these kinds of issues on particular health topics. And I think that's something that didn't exist when I was going to school and training. And so I think if you want to learn about those things or find out about future opportunities, keeping an eye on those things in real time, conference on, blah, 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 you might find some of those things that way as well. For example, I missed it, but there was a conference on AI and developmental disabilities earlier this week, sponsored by the American AUCD, which is one of the disability organizations. So just as an example.
Very good, thank you. All right, next question about AI. And you have answered some of this in your presentations, but if you would like to elaborate on it a little bit more, this is an opportunity. How do you see AI and machine learning influencing the future of health informatics, especially in your expertise, areas of expertise?
Yeah, definitely. So, especially I see it affecting real-time data. So, like faster lab results and X-ray results, and that actually would lead, if it's accurate, would lead to very good results because we wouldn't have patients wait for their lab results. So it would definitely also allow for early interventions. And another thing is it would help in the automation as well for data quality and curation. So yeah, I can see a lot of importance for it if it's also done well and ethically.
Thank you.
Yeah, I know the wave is coming and it will both do great things and knock people down. So I think, you know, I'm a little more skeptical of where AI is currently, although I realize it's moving fast to actually change recommendations and practices in a way that is truly empirically based and well validated. But I have optimism that could get better, as long as it's done ethically, as Luna said, and also as long as there are systems incentives to do continuous evaluation and improve things over time. I worry a little bit that when we get something that works, we'll just go, oh, we got it. And I don't think that's really the way this stuff works. I will say one of the areas I'm excited about AI is the ability of AI to lower barriers to healthcare. So, for example, when you get your post-visit summary that you get in your electronic health record, or sometimes it gets printed out for you. That's the kind of thing that many people don't read, regardless of whether or not they have a disability. And if they have a disability, it may be in a format that's very hard for them to read. And I think AI can both translate and simplify information into plain language for people with disabilities, particularly intellectual disabilities, and also create ability to have interactive tools where you could query a question, like, what did my doctor say about this? And then it will tell you what the discharge summary said. You don't have to find it in it. Or similarly, I want to tell my doctor about this problem I'm having. An AI tool can help you figure out how to tell your doctor about it in the way that's most likely to get the most results or get the tests lined up before you get there, things like that. Or wearables, digital devices, as was mentioned before, about how we can incorporate that data to help lower barriers to health access and things like that. So I'm very excited about the way AI can potentially improve accessibility. But I do think we have to be mindful of the errors and the fact that there's sort of a confirmation bias effect of like, oh, look, isn't this wonderful, without actually looking to see whether it's right or wrong. And there have been countless examples of that in public spaces where people do things on AI and think it must be right, and it's not. A lot of the time it's only as good as both the data and the tool. And we have to have ways to look at exceptions. AI may tell us that this is usually the way it is, but there will always be, science and medicine are replete with examples of times where there are exceptions to the rule, and that's never more important than in the care and treatment of an individual and their health problems. So
very good, thank you. I had kind of a question on ethical and societal challenges, but I feel like both of you answered that. I do have an audience questions. What is the most impactful piece of advice you received for your data career? Either one of you.
Luna, you want to go first? Or you can think and I can
Yeah,
bring it.
I'm trying to think. I received a lot of advices, but I think maybe one of them is always validate your data science steps with your team member, because we always tend to like as humans make mistakes and we don't see them, we disregard them. But if we have someone else that checks and always like run another check is important, especially in the medical field. So I think yeah, that's one of the advices.
Yeah, I would echo that. I'm not sure I ever got it exactly as a single piece of advice, but I think skepticism about every result you have, and then building in sort of like Malcolm Gladwell's, or not Malcolm Gladwell, I can't remember, Atul Gawande, checklists, building in checks that check our biases. There's lots of data about implicit bias in individuals and how it affects our judgments of things, but that's absolutely true in data science too. So, and then also what question does your data make you want to ask next? And realizing that there's always got to be a next question, and being critical about that, and also not being, I think, a lot of science is about failure. Some of the most important discoveries, whether it's in data science or other things, have come out of things that didn't work out the way you thought they would. And so also looking at failures as opportunities.
Very good, thank you. And there's two more questions about getting into data science. I will save those questions and email it to you, and you can follow up with the askers as appropriate. All right, well, we are almost out of time. I will tie this up with just looking into the future. One last question. How do you envision the future of women in health informatics? And what can we do, or how can the next generation prepare to be leaders in this space?
Well, I can start, although I'm not sure I think of myself as a pure data scientist, but I think, historically in science, and in medicine, women, in part because there have been barriers to women's full inclusion and participation that still exists today, have often been people who are in positions to ask innovative questions, and do innovative things. Sometimes because no one else thinks it's the hot thing, and sometimes it's because inclusive workplaces lead to framings of different questions and perspectives. So I think that's an opportunity for women moving forward. And I also still think that we're working towards a world where there aren't biases towards women in more data science oriented fields. So I think in that way, it's an exciting time potentially for there to be more equity and opportunity.
Thank you.
Yeah, I echo that, and also would add to it, like for the next generation, to always ask who might be left out, so that because true innovation in medicine comes when we design for everybody. So I feel that's very important.
Thank you. Thank you so much. I would like to thank all three of you, Cynthia included. So Holly, Luna and Cynthia for being here with us today and participating in this very engaging and fascinating panel discussion. I appreciate your time and that you came and shared your expertise and thoughts on the topic of women in health informatics and data-driven medicine. We are right at 11 o'clock, and we have our next presenter, Dr. Cecilia Lee, already online. I was wondering if we need to take a 5-minute break, or if you would be ready to go. I don't know if I just put her on the spot. There she is. There you go. All right. Do you think we need to take a 2-minute, 3-minute break, or are you ready to go, Dr. Lee?
Oh, are you asking me? Oh, I thought you were asking the audience. And I said, I'm not, I don't mind either way. So I'm happy to go if you are ready or.
All right. Then let's go ahead and get started.
Okay.
Thank you. Thank you for being here today. And I have the honor of introducing you, Dr. Cecilia Lee is the professor of ophthalmology and a Klorfine family endowed chair at the University of Washington, where she also directs clinical research for the Department of Ophthalmology. A clinical epidemiologist and retina specialist, Dr. Lee's research bridges ophthalmology, data science, and neurology, applying big data and artificial intelligence to uncover early biomarkers of systemic and neurodegenerative diseases. She leads the NIH-funded Eye Adult Changes in Thought, also known as Eye-ACT study, integrating retinal imaging, brain MRI, cognitive testing, and electronic health records to better understand aging and dementia. Dr. Lee also participates in national initiatives such as the NIH Bridge to AI program, focused on generating AI-ready dataset for salutogenesis in type 2 diabetes research. Dr. Lee is committed to advancing collaborative, data-driven approaches in healthcare research with the goal of improving early detection and patient outcomes across aging and neurodegenerative diseases. Please help me welcome Dr. Cecilia Lee.
Well, thank you so much. I'm sorry, I was attending another conference, so I couldn't attend the whole meeting today, but I just joined their last bit of conversation, and I'm super excited to give this presentation at this audience, because there is definitely an intersection of research, AI, ethics, and data science. So hopefully the presentation that I'm going to give will provide some areas that we can discuss further. Okay, so I'm Cecilia Lee. I'm a medical retina specialist at the University of Washington, and I'm a clinician scientist. I do about 75% research, and my research is focused on big data, AI, clinical epidemiology. So I'm going to get started. So my presentation is going to give a brief overview of big data, how we think about and how that might have changed the way we design research these days. I want to give an examples of how we could probe the brain through the eyes using leveraging this big data. And also, I want to share our experience in doing AI-ready data collection and sharing via AI-ready project that I'm going to present, and I'm going to end it with future directions. So what is big data? So I'm sure you're very familiar with what we call big data. Sometimes it's called as 3 V's, as big volume, velocity, and variety, and sometimes 4 V's with validity or variability included. And I think of it as big data as many large-scale data sets that are difficult to analyze with traditional statistical methods. So yes, you might be able to fit it with R or SAS, but it's really not the actual statistics, but it's actually visualizing the data set as a whole for you to even engage, well, what type of analysis should I run, or what type of what type of data am I focusing on? So I'm hoping that I can share that with you. The way I see data science and big data, I like this figure because we know what artificial intelligence is, and then machine learning is a part of AI. Deep learning is part of machine learning. LLM that we are so aware of these days is part of deep learning. And so big data, I like big data, how that is actually the focus or the nidus in which really help the AI field to advance forward, because now we have to have a good quality large big data to train sophisticated models these days. So why do I say that new paradigm is in medical research with big data? And I'm going to give an example in ophthalmology world. So in the eye clinic, routine eye clinic visit today, we obtain, and then I'm going to see if I can make the laser pointer here. Can you guys see the laser pointer or some kind of spotlight?
Yes.
Okay, great. So this is an example from this photo, and then the IR image that we obtain. I'm sure if you have gone to optometry or ophthalmology clinic these days, and they have really taken these kind of photos. We take a look at your eye with the slit lamp photos, and then, oh, this is an image of like OCT, and then, so all this imaging database, which, for some imaging like OCT and geography, it's 2 to 5 megabytes per person. And then that kind of data, along with electronic records, they all go to big data pipeline. And then these are all being passively collected during routine clinical care. And so we are living in this passively obtained big data pipeline that allow us for us to take, a look. And then that becomes our research database already, even if you didn't quite design what the study was going to be about, what data to be collected. And I'm going to share some of the advances that have already occurred. There have been so many advances in ophthalmic imaging technology. So this is an image of optical coherence tomography, which is, the laser, takes a picture, and then we get an image like this. This is an OCT angiography where the level of capillaries, at the level of capillaries, we can see this blood, blood vessels, and this does not require any dye. So it's amazing what we are able to obtain in a matter of seconds in less than a minute. So this is an image of that is an image of fundus photography. So this is the a fundus photo of the left eye. This is an optic nerve. This is the vessels, and you can see a lot of kind of blood. Those are hemorrhages, and this patient has diabetic retinopathy. And on that photo, you can kind of tell that, it would be hard for humans to like label everything that is abnormal. And this is an image of AI algorithm that is segmenting and quantifying all the abnormalities. So you can imagine how sensitive and specific we could get with the quantification of data that was rather subjectively interpreted before the era of machine learning. And this is an image of an optical retinogram, ORG. This is at the very research stage, but we now have the ability to actually stimulate a single photon, a single photoreceptor in the retina, and then detect its response. So we can be pretty specific and pretty sensitive in terms of what we want to obtain. So what is a big goal? The goal of big data is that it's not just the EHR record, right? We are really trying to combine all the large data sets, such as genomics, metabolomics, medication data, but not just that, but behavioral and environmental data to truly understand what are the genetic and then environmental risk factors for somebody to develop a disease and progress. So we are trying to, come up with an early risk prediction, coming up with better differential diagnosis and optimized treatment as, to improve the personalized medicine aspect of medicine these days. So that's the big goal. And then that's what we do. I co-lead competition ophthalmology lab with Aaron Lee. This is this is what we do in terms of doing data analysis using lots of different types of data sets. But one thing that I'm passionate about is how big data really allows to connect novel biomarkers. That big data in a way that was not possible maybe 20 years ago. Even when I was a medical student, this was not possible, right? I was one of those medical students that were going over all the medical charts and then, kind of putting it in the spreadsheet, who had what, and then, I had to go to another clinic and then pull all the data to try to compile everything. But now with the advances of big data and machine learning approaches, it really allows us to connect the dots in a very efficient way. And then I think that will lead us to a new path in terms of where to do research, where to focus on understanding the better of the disease. So today I'm going to focus a little bit on how we have been able to connect the dots between ophthalmology and neurology. So more than 46 million people live with Alzheimer's or other forms of dementia in the world. And every 65 seconds, someone in the US develops Alzheimer's disease. And in And then we know that in 2050, this number will almost double. And Alzheimer's disease is the only leading cause of death, the top 10 leading cause of death that really does not have good preventative or, definitive therapy yet. So we are using the concept as the eye as a window of the brain. And I feel very fortunate that I am an ophthalmologist because the eye is actually a part of the brain. And then during the embryology, within the first month of embryologic development, the eyeball actually comes as an outpouching of the forebrain. And then the actual retinal layer is the neural layer of the brain. So we can learn a lot about the brain just looking at the eye. So I just mentioned that the eye has the same embryologic origin, has similar vascular properties with the brain, and the evidence from clinical studies are pretty convincing that we can know a lot about the brain health by looking at the retinal health. And then these are some of the images that I just showed you a little bit ago. And then these are very accessible and scalable. So, we think that if we know that the eye diseases alone are predictors of Alzheimer's disease or dementia, now we can have this, beautiful machines that can obtain very sensitive visualization of retinal health. Then, can we actually tease out what are the retinal features that are predictive of cognitive decline or other brain or other systemic risk factors. So that is our goal in our lab. So we have been going at discussing kind of how do we really apply deep learning and artificial intelligence and big data to understand these concepts. So one of our first study was really looking at, are the eye diseases predictive or are they associated with future risk of Alzheimer's disease development? So for this, I need to give an intro to Adult Changes in Thought Study or ACT study. So the Adult Changes in Thought study started in 1994 in Seattle. It started with group health that now became Kaiser Permanente Washington. So these patients are actually members members of Kaiser Permanente. So they get all their comprehensive medical care through Kaiser. And if they were 65 or older, they were randomly drawn and then invited to attend the screening meeting. And if they were cognitively normal at that point, then they were enrolled, and then every 2 years they receive a multiple battery of cognitive tests. If they went below the borderline of the normal cognition, they underwent even more tests, including physical exam and whatnot, and then they received a diagnosis of either Alzheimer's or other disease, other dementia, by the consensus criteria grading methods, meaning that that is the state-of-the-art clinical diagnosis of Alzheimer's, meaning that multiple experts from different domains meet, and then they discuss the case, and then they come up with probable or possible Alzheimer's disease. Because as you know, we actually do not have a definitive diagnosis of Alzheimer's disease until we have the brain biopsy. And so the beauty is that this has started as 2,000 people maintenance cohort in 1994, but then now more than 5,600 people have contributed their time into this data set. And then now we're hoping to increase the maintenance cohort from 2,000 to 3,000. So we're hoping to get even more data as we go longitudinally. And we have over 1,000 brains that have been already donated and autopsied. So again, they are part of prospectively collected dementia-free participants. They are enrolled, they do every 2-year cognitive testing, and then they are followed until they either develop Alzheimer's or their dementia or they pass. But the beauty of this study design is that because they are part of this HMO, Kaiser Permanente, we are able to access all their medical care. So, and then some of them are pretty long, and then I'll show you an example. So even though the looking at the eye information was not part of the initial thoughts or initial purposes of this study, we were able to access comprehensive ophthalmic clinical data. So our first question was, well, are these 3 age-related conditions? So these are an example of age-related macular degeneration, or AMD, glaucoma, or diabetic retinopathy. Are these associated with future risk of developing Alzheimer's disease? So we had data from 3,900 participants, about 31,000 person year, and then almost 800 Alzheimer's disease incident cases. So this is a forest plot. So we know that if it's right in the middle of hazard ratio of 1, then there's no risk. If it's more than 1 in the red side, then that is associated with higher risk of Alzheimer's. So we know that age is associated with higher risk. If it's to the left in the blue side, then it's protective. So we know that education is protective against dementia and Alzheimer's. So ApoE4 allele is the highest or the most well-known genetic risk factor for developing Alzheimer's disease. And in our cohort, it always is about 60% more risk, higher risk. So it's like right there. But what we did not anticipate was that if your eye looks like this, which is a typical example of AMD, then your risk was definitely higher for developing Alzheimer's disease. Your risk of developing Alzheimer's disease was definitely higher if you had diabetic retinopathy, and then also early glaucoma. And this was, made us really happy and excited. But actually, what was really surprising was that the degree or the severity by which the each eye disease was associated with was really surprising. The fact that they were all kind of like aligned here means that me, by taking a look at your eye, could be as good as knowing this blood test and detecting whether you have ApoE4 or not. So this kind of really gave us a foundation that yes, there is actually a lot to know about the brain just by looking at the eye and then exquisitely characterizing the health of the retina to better understand the the mechanism and potential treatment of Alzheimer's disease. So that was our first paper. Then the second study that I want to show you is is visual function actually an indicator of future risk of Alzheimer's disease and dementia? So I had mentioned, the study design, and again, they're enrolled and then they get, regular cognitive testing. But throughout their membership of Kaiser, they have gotten many eye exams, right? And then what we did was that they underwent this transition from paper to EPIC in 2003 to 2004. So we developed our in-house natural language processing algorithm to obtain all the feature and then visual acuity and visual function data from EPIC. But we wanted to go beyond that because we really wanted to leverage all the longitudinal data that exists. And we went as far back as 1940, especially in people who have gotten autopsy or who are consented to the autopsy. So you can see that this patient was actually 29 years old. And then they, they had to be at least 65 or older to get enrolled into ACT study. So this means that I have at least 40 years of history on this participant. So, but you can see that the handwriting is awful. So I had to, an army of manual extractors were constantly emailing me saying, what is this handwriting saying? And then I'll just be like, I have no idea. It just looks like a chicken scratch. But so we did it. And then so I'm very excited to show you our results. So we had about 9,000 eyes from 4,500 people. And then we have divided into the eyes that never develop dementia versus Alzheimer's disease versus non-Alzheimer's disease dementia. And in our case, most of them are vascular dementia. So in summary, we actually had about 250,000 of encounter data. So this is the work of Aaron Lee. And then, I have divided these 3 panels. As the first panel is the eyes that never develop dementia. The second panel is the eyes that develop AD. And then the last panel is the eye that develop vascular dementia. So let's see what it looks like. So each graph, each line is one individual. So you can see that you can appreciate this fluctuation that exists, as you get older. And then, sometimes you have good days, sometimes you have bad days. Sometimes, maybe you've had cataract surgery, but, this is what I mentioned earlier, the visualization. Visualization of the big data is key for you to get the sense of like how messy your data set might be, and then what kind of conclusion can you derive from it. So all I can tell is that, yeah, as you get older, it's amazing how you started from like age of 25. As you get older, it does tend to get lower, meaning vision gets lower. But what do we see? Out of about 155,000 years of follow-up, what we did was that, so this is a summary graph. This is the eye that never developed dementia. This is the eye that developed AD dementia. And then this is the eye that developed like vascular dementia. So what we see here is that clearly during early life, they're all over together. There's no difference. But between this age, there's a clear inflection point in which people who will actually eventually develop Alzheimer's disease will start seeing less vision, decreasing vision compared to people who have no dementia. But what's even more interesting is that this inflection point against dementia, against healthy controls, is that the non-AD dementia group actually differentiates even sooner than this inflection point with Alzheimer's disease. What does that imply? I think that implies that these people who have vascular dementia, they probably have diabetes, diabetic macular edema, or maybe conditions that actually impact the visual acuity earlier due to systemic diseases. And then so that makes sense that the visual acuity gets down a little bit sooner than people who develop Alzheimer's disease. So this is very interesting. And then it also kind of supports this idea that late life vision loss is a risk factor for developing Alzheimer's disease. And if we were to do something, then that should be helpful. And I'm going to show the next study to kind of advocate for that. So out of all this data, we are showing that visual acuity trajectory alone can predict AD dementia risk. So when we talk about, this type of, visual acuity or, eye diseases, it's a new risk factor that we just, discovered. And as an epidemiologist, you are very happy to discover a new risk, but you're even happier if it's a modifiable risk, right? So then we asked, well, what if we intervene? What if we do something to increase the visual function or to increase the eye health? Does that impact the future risk of Alzheimer's disease and dementia? And so we asked, is cataract surgery associated with risk of developing dementia or Alzheimer's disease? And that was because, cataract is a very aging-related condition, and then you need surgery eventually. And we wanted to know that, does that intervention actually help, or can that protect against the development of Alzheimer's disease or dementia? And so this is a flow diagram. I'm just gonna say that we did Cox survival analysis, and we used surgery as a time-varying exposure to kind of not have this like immortal time bias. So what do we see? So I'm going to show the forest plot again. So the top panel, the outcome is all-cause dementia, and then bottom panel is the Alzheimer's disease dementia. So if they did not have any ApoE4, then it was clearly protective. So hazard ratio was about like 0.5 something. But what was really exciting was that if you had cataract surgery, then the hazard ratio was about 0.7. So there was a 30% risk reduction in people who developed who underwent cataract surgery. And what was even more exciting was that this significant difference was detected even after a decade following cataract surgery. So I think when I just heard somebody say that, always validate or always kind of question your results, especially when results are good. So then I actually did drive my biostatistician pretty crazy, because we were trying to do all the secondary analysis to try to control for potential bias. Luckily, we had a lot of research criteria like survey results, so we were able to kind of do that. But one of the key bias control that we did was compare this to people who underwent cataract surgery versus people who underwent glaucoma surgery. So cataract surgery is an outpatient surgery and it's an optional surgery. So you would not consider having cataract surgery if you have like congestive heart failure, or if you have like a lot of medical conditions that you need to take care of. And then so we thought that, well, glaucoma surgery is also kind of like that. It's an outpatient surgery, so you need to have certain physical fitness to be able to undergo this surgery. And in contrast to cataract surgery improves vision right away, but glaucoma surgery, the goal is not to improve vision. The goal is to actually stop the progression of glaucoma so you don't keep losing more vision. So it's very different. And then so we compare that. So we compared to people who had glaucoma surgery to really control for this healthy patient bias. And what do we see? So this is the hazard ratio. So people who had cataract surgery, what was the risk of developing dementia? And again, it's hazard ratio 0.71. So it was about 30% reduction that I showed you. And if we looked at model 3, now it was on people who had glaucoma surgery, what was their likelihood of developing, Alzheimer's and dementia? The risk was null. So that really gave us more reassurance that this was not just about the person being healthy enough to have an outpatient intraocular surgery, but it was more specific to somebody having had cataract surgery that improves visual function. So there are many hypotheses. Obviously, if your vision is pretty poor, then now after cataract surgery, your world is so much crisp and sharper, then you're going to be happier. You're going to want to go and become more active. You might be able to become more social, or take on a habit that are intellectually stimulating, all of which are known to be protective against Alzheimer's disease and dementia. But there's this impact of improving the stimulus between the neurons of the brain, and then also decreasing overall cognitive overload, because your brain is not constantly thinking or spending way too much time to try to figure out what it is that you're seeing in the world. So all these kind of papers that I just summarized were included in this recent Lancet Commission report from last year, in which 2 new factors were found as 2 new modifiable risk factors, and then that included late line vision loss. So we're very excited, and then I hope this kind of illustrates how big data can kind of really find a new link or new biomarkers or new risk factors that can improve our understanding of the disease. So that's the vision loss right there. So I'm going to change gear a little bit. So I had a focus on big data. And then earlier on, I said that it's really this marriage between big data and artificial intelligence, because artificial intelligence needs really good large-scale big data to train. But then to train big data, you need more sophisticated, powerful artificial intelligence models. So this kind of gives rise to Bridge to AI program. It's an NIH-funded program that initially has recognized the power of AI models in biomedical research, but recognize that there is no good big data that is AI ready for training models. What do I mean by that? So a lot of data sets that are available currently to train machine learning models happen to be very biased. So one bias could be, demographic bias, right? Maybe it could be, mainly on white population, or it could be mainly biased towards either healthy people, like in the clinical registry or UK Biobank, or mainly biased towards sick people because you are, leveraging EHR data. And obviously, if you are not sick, then you don't happen to go to, doctors' offices. So Bridge to AI really asks for the data generation project to collect data in such a way that is ready to be used for machine learning models training. So going back to, where are we with machine learning and deep learning? So I would say maybe a decade ago, we were very excited about deep learning and then the way we started using deep learning in the medical research was, initially classification. We did the supervised learning where, we trained the model say that this is a cat, this is not cat, this is cat, this is not cat. And then so the machine would spit out saying that, yes or no. If we added localization, the model said, not only it's a cat, but it's here in the center. You could do object detection that there is cat, dog, and duck. And then instant segmentation was that not only there are multiple animals, but then where, and then you could segment the edges, and then you can kind of see how that would be really helpful in analyzing or interpreting medical imaging. But what was even more exciting was that there is this unsupervised learning. So we don't quite know how it learns, but this is an example of deep learning model predicting where is horse and then where is a zebra. And it's kind of interesting how the model already knows that zebra, goes into the brown background as opposed to horse goes to the greenish background. And what is even more surprising is that there is newer models for this, where the model is now without any training, any previous training data or any previous training, it is immediately recognizing what the main message of the data of the video is, and then it can discern the background from the main photo. So this is kind of pretty unbelievable and amazing, and that this was published a couple of months ago. And what this infers is that now deep learning can organize information de novo without the guidance of clinicians. So we are kind of moving away from, we're advancing from us clinicians having to tell deep learning, this is what I want you to do, this is the examples, this is the training, and this is what you do. Of course, that's helpful. That can make our human efforts a lot more efficient and helpful. But then now this kind of field is really helping us to get new insights that were maybe hidden to our naked eyes. So going back to Bridge to AI, so we are right here. So there's a data generation, data preparation. So we know that, out of all this, understanding the AI ecosystem in the research of biomedical science, we know that the first step is really collecting the data that is quote unquote AI ready. And I'm going to explain what we mean by that. So I mentioned that there were 4 data generation projects in the country, and we are the Salutogenesis group. And Stanford is also part of our site, so we are very excited. And I'm going to explain a little bit more about the goal of AI Ready or Salutogenesis Grand Challenge Data Generation Project. So our goal is to create a multidimensional, ethically sourced data set in diverse people for studying salutogenesis in type 2 diabetes. It's pretty mouthful, and then we call ourselves AI Ready. Oh, okay. So okay, so we are very used to understanding what the pathogenesis is, right? We are very used to, discussing risk factors. What are the environmental, genetic, and behavioral risk factors? And salutogenesis is the opposite. How do we become, how do we become healthier? So it's actually the exact opposite of pathogenesis. So how do we envision us using this data set to understand salutogenesis? It's borrowing the success of single-cell RNA-seq world. So in single-cell RNA-seq, at one time of single-cell RNA-seq, you get amazing tons of large data of each cells. And there's machine learning technique called pseudotime manifolds, where the model puts all the cells that are very similar to each other right next to each other, and then all the cells that are kind of very different from each other very far away. So if you actually put them all together into pseudotime, manifold timeline, you can actually visualize the cell progression from totally embryonic stage to totally differentiated stage. So even though you obtain this data at a single time point, you can kind of generate this pseudotime atlas of cell progression. And our design was saying that even though we are collecting this lots of multimodal data from a large number of participants at one time point, if we are able to leverage this technique, then you will be able to tell this pseudo-time atlas or history of somebody becoming more disease or healthier. So the design of our study was to really get lots of multimodal domain study data collection to enable development of cross-sectional AI models, and then also predictive AI models using pseudotime manifold techniques. So we are collecting 4,000 participants from 3 sites, from University of Washington, University of California, San Diego, and University of Alabama at Birmingham. We are balancing the cohort. So even though 4,000 does not seem like a lot, because UK Biobank has, 500,000 people, but the key is really balancing the severity of the disease, because that really gives you the power to train sophisticated model with small number of data. So we are balancing 1,000 people into non-diabetic, 1,000 people who have either diet controlled or preclinical diabetes, pre-diabetes, and 1,000 people into medication controlled, and 1,000 people insulin controlled. And we are balancing on Asian, Black, White, and Hispanic, and then female and male biological sex. So we go from head to toe. So even though we are ophthalmologists, eye is actually a small portion of the data collection, because we really wanted this to be understanding salutogenesis and pathogenesis of type 2 diabetes development and progression. So when they come, actually before they come, they do lots of surveys like like health history. What is your diet? What is your diabetes related, daily life, daily activities of living, and then social determinants of health. We use Phoenix surveys, and on site, we go over all the medication. We do vision and retina imaging, blood tests, vitals, and then we get a lot of blood and urine testing. We do 12-lead EKG, MoCA, and we do monofilament tests, and then we actually obtain biospecimen for future basic science research and even the PBMCs. And after the site visit, they go home with 10 days of continuous glucose monitoring, 10 days of Garmin fitness watch, and then 10 days data collection to be done by environmental sensor. And I'll explain more about the environmental sensor. So this is an example of all the retinal imaging they go. And one of the difference or one of the key strengths of our project, I think, is that we actually don't hold any of our data to ourselves. We actually, none of the investigators will actually publish data before the data becomes public. The only exception is the interns that we train, because they help us to do QC, and then they are part of doing research with our data. And every year we release our data so that it's open to public, and then they can use, they can really take advantage of all this, data that we have, collected. So in year 2, we released little over 1,000 people's participants' data, and it's enormous data. So we have more than 1,000 people's standard 12-lead EKG. We have more than 1.4 million oxygen saturation measurements, near 3 million glucose measurements, and then we have 182 million air pollution and light spectrometer readings from the environmental sensor, and then these are kind of one of the largest, if not the largest, retinal imaging data set that is open to public. And so this is knowing that our goal is 4,000. This is only 1/4 of our study population, and it is 2 terabytes of data. And by last month, near 450 research organizations worldwide have downloaded our data, and then we are, very excited that people are able to take advantage of this. So this is an example of how are we doing with the balancing. Okay, so this is looking at the type 2 diabetes status. So I said that our goal is really to balance the spectrum of the disease. And then this is a decade of life. So we have a pretty balanced, age spectrum out of those 4 groups of diabetes. And then now I'm going to show you the sites from UAB, UCSD, and University of Washington. And then we even provide the split that we suggest for deep learning or machine learning models so that there is no confusion in, if somebody wants to validate somebody else's results. And then there are virtually no missing data. So this is a missing ECG. FLIO, this is another research data. So this one actually does have more missing data, but then the more standard retinal imaging data, there's really no missing data. So that's the how we are doing with the balancing. I'm gonna try to advance here. Okay, and then how can you access this AI ready data set? So we have created our new platform that you can access the data set. Now you might wonder, and then we have done a lot of work keeping this like well documented in terms of what is this data set about. We also created a license that is shared more openly, but then it specifically prohibits somebody from try to attempt to re-identify people using our data set, and then also to try to harm any particular populations, because we are worried about minority population that might be that some people might try to kind of study and then in a harmful way, but this allows any commercial product development and whatnot. And we allow, we provide any training material if somebody does not have a human research training, and then these are for free. And then we have documented lots of health sheets so that you know exactly what you're getting. And then we develop metadata and example outputs for each data variables. So this is an example of how we try to make this, our data, quote unquote, AI ready and machine readable. So I am, do I have some time to kind of go over some examples, or I am happy to kind of finalize more so that we have more time for discussions?
You have some time, yes.
Okay, all right. So I'm going to show really quickly some of the potential applications using our data set. So I had mentioned that we have developed an in-home home environmental sensor. We have always been interested in looking at, what are the impact of, pollution or that have on eye health or any systemic health. But we've noticed that most of, studies in the literature have focused on outdoor condition at a broad city or regional scale. So we wanted to create a home-based sensor that, they can take it home and then they put it in their home and then just like plug it in and then just leave it for 10 days. And this home sensor collects every 15 seconds all the particulate matter counts like PM 2.5 and temperature, relative humidity, volatile organic compounds, and then also 11 multispectral light sensitivity measurements. So it looks like this. It is less than 2 iPhones together, and then it does weigh less than your iPhone. And so what can you get? So this is an example. This is a work of Jamie Schaefer from our lab, and this is a comparing patient A from patient B environmental sensor data. So participant A clearly had lots of PM 2.5, and participant B really did not have any PM 2.5 at their house. And then also you can kind of see that there's a difference between VOC. And then so it kind of gave a reassurance that, yes, it is actually sensitive to detect differences in this exposure between individuals. What was really surprising and then really made me excited is that this idea that maybe environmental pollution is, could be a trigger or could be impacting, how severe you are in the diabetes. So this is PM2. This is a mean max of PM2.5. And then this group is no diabetic group. This one is pre-diabetic group, and this group is non-insulin controlled, and then the other one is insulin controlled. So there is signal that there is higher pollution level at the households of people who have, either no diabetes or diabetes, but there's a kind of dose-dependent effect between diabetes severity and air pollution, which is really exciting. And this is a volatile gas mean in 75 percentile. And again, there's that small, but there's a dose-dependent effect. So we're excited that to kind of get more data. So in year 3 data, we should have about like 1,500 people more being released. So we're hoping to see maybe stronger signal once we have more data. So this is an example of like big data providing a new insight or new research direction. And I wanted to kind of mention, why are we so interested in retinal imaging? Obviously, we are biased, but retinal imaging is really providing a non-invasive way to kind of make you understand what is in vivo, the histopathology of the structure and the vessel of the retinal imaging. So going back, so this is an eyeball from the cross section. This is an optic nerve and vessels, and then these are an OCT, optical coherence tomography, that can be obtained in 5 seconds, and then this is like a cross-sectional image of the retina. And as you can see, that these retinal layers are looking just like histopathology slide that our pathologists would look at. So now we can kind of differentiate not only whether it's swollen or not, we can even look at, is there thinning of what layer? And then is there, fluid or blood in which layer? And then so that's an example of how sophisticated we can get, and then how well we can characterize the health of the retina. And OCT angiography is an example where, I had said that you don't need any dye, but we can get the information at the level of capillaries, which is amazing. And then this is an example that we published many, many years ago. So this is an OCT from people who have no diabetic retinopathy. I'm sorry, the very minimal to no diabetic retinopathy, people who have moderate diabetic retinopathy, and people who have severe or proliferative diabetic retinopathy. And I'm sure you can appreciate that even, just looking at it, they have capillary dropout, there's an ischemia, and then this is like the quality of OCTA that can give us, information on how much like vascular disease they have. And then I hope you can agree that this can actually be helpful in understanding better systemic cardiovascular health, and then maybe even the brain health. So am I kind of going, is this like a far-fetched idea that from the retinal imaging, we can understand like systemic health? So this is a paper that came out from Google's team like a couple of years ago. And it was amazing how they're seeing that out of fundus photo, the machine can tell us the exact age and gender, and then whether they're smoking, hemoglobin A1C, and BMI. I'm a retina specialist. I look at the fundus photo every day. There's no way I can just, definitively say, what is the age of a decade or sex. And it's pretty remarkable that the machine was able to even like estimate systolic blood pressure and diastolic blood pressure. What makes me think that there are a lot of latent, hidden features or biomarkers or latent information that are embedded in this imaging that we as humans just cannot see. So I'm hoping that as our data set increases and improves in quality, and our models and ability to detect and find the association through artificial intelligence improves, we are able to learn a lot more about, our health overall. So I'm gonna just finish with one last paper. So this is a paper that was published in Nature, and our lab was, part of training this model. So this is a medical foundation model where, they use the retinal imaging to use the, can you do like ocular disease progression prognosis, which is not very, if you think about it, it's not a very hard task, right? Because you use the eye data, and then now you're using to predict, whether they have the diagnosis or they're going to progress worse. But what was really kind of intriguing and exciting was that using the eye data, they were using to predict what is their 3 to 5 year incident rate of developing ischemic stroke, MI, heart failure, and Parkinson's disease. And what is amazing is that they actually did pretty well. I mean, there's no way I could say that, sir, with like 60%, accuracy, I can tell somebody whether they're going to have a stroke in next 3 to 5 years, or Parkinson's disease, or heart failure, that was like 70%. And what it was trying to show that not only that, but by use of foundation model, you have a lot less, you need a lot less of a training data. So you no longer need millions of data. You can see that the AURC curve really did not change much when you're using 20% of training data as opposed to 100% of training data. So we are really getting better at training, exciting models. So to summarize, this is our marker paper in terms of how we designed and what we are envisioning for our AI Ready project, which is published in Nature Metabolism. And this is our protocol paper in terms of if you wanted to look at specifically at the protocol. And what are we doing now? So we have two sets of data set that we are sharing. One, you can actually go online today, and then you can download your data set after you have gone through accreditation, but you don't need DUA or anything. But there is a control data set that will need DUA, so we are in the process of finalizing DUA and then data access committee for that. And then we are running the first batch of whole genome sequencing on 2,500 samples this year, and then we're hoping to do that the rest of them next year. And the year 3 data should be released by December 1st, and then that will provide us about a total of 2,500 people's data. So take-home points: big data and AI approaches have potential to unlock novel connections, risk factors, and hidden disease trajectories. But understanding limitation and validation with other data sets are critical. But collaboration is key to unlocking potential. So thank you so much, and I want to thank the participants and our study team and our lab members, and I'll take any questions. Thank you.
Thank you, Dr. Lee. That was eye-opening. I made that joke. There is I do have an audience question, 2 of them. Have you worked on data where someone had by birth eye defects, like squinting in relation to dementia?
So, okay. So in terms of dementia, so if you're referring to ACT study, then there was no exclusion criteria. So they could have been blind, but there weren't that many people. We actually did want to look, because is it really the blindness that are from birth, or is it that you were not blind and then you became blind, and then that is what increased the risk of dementia? Per literature, and then I think it intuitively makes sense that it is the later life visual impairment blindness that you get that increase the risk, because if you're born blind, then really the cognitive overload should not happen, because the brain is plastic enough that brain is relying on other senses.
Very good, thank you. And there is another question relating to AI. What strategies are you employing to ensure that AI applications in ophthalmology are both effective and equitable across diverse patient populations?
So I think not just in ophthalmology, but in medicine. So let's just like go with the example of AI already. So this was really our way of try to make the data balance, because we know that a lot of bias from the models come from the bias in the study data set, right? So that's why we wanted to divide it in equal amount of not only the disease spectrum, but the demographic, and then also, sex. Obviously, that is not enough. And then that's why we are obtaining a lot of, social determinants of health data. We would love your help in analyzing what we have got. So note that our data is really to generate data for the good of all the researchers worldwide. And then, we don't have all the expertise. So I've been actually looking for people who can help us, understand, how do you analyze all the social determinants of health? Like, what is important, and how do you kind of really put that in perspective with all the clinical other data that we get?
Thank you. Any other? I don't see any more questions. Last chance for the audience to ask. I will give it a few seconds. But in the meantime, I did want to thank you for closing out the conference and sharing your expertise and incredibly interesting research. And also about the AI Ready project. And if we don't have any more questions, I don't see any more questions, so I will go ahead and wrap up the conference. I would like to thank all of our speakers and panelists for being here today and participating in this conference. I am personally very inspired and heard a lot of very interesting career journeys and research efforts that women are currently participating. I wanted to apologize for the technical issues in the very beginning with the two videos, the short videos that I was trying to play, and there was no sound. You will receive the recording and the slide deck, and then you will be able to enjoy those two introductory videos. And lastly, I would like to thank my team here at Lane Medical Library, Danielle, Amanda, Colleen, and Connie, for all of your help with putting this conference together and making it being a success year after year. I do have a quick slide if you would like to provide feedback. This will also come in through the follow-up email, but if you want to quickly scan the QR code, you can definitely do that. There's a quick feedback questionnaire, a survey, less than 2 minutes to fill it out. It will help us plan next year's event by learning what we did well or what we could improve on during this year. I want to thank everybody for being here today and participating, asking questions, listening. Hopefully you learned something. I will stop the recording now, and thank you so much and have a wonderful rest of your days.